question, maybe the same we can expect in INICET also. And biochemistry is one of the subject where one can score maximum. The reason is there are very repeated topics, the repeated questions that is commonly asked from the biochemistry part. But the problem with the subject is we find it that there are so much of data to be remembered, to be crammed. Plus, secondly, the subject feels very dry. There are so many cycles are there, and we feel that how we are going to remember it and what is the clinical relevance of this subject is. So we will how we will proceed is we will try to connect as much as possible with the clinical side so that it becomes interesting for us, and that also is going with the trend that this is how they are framing the question, connecting with the clinical subjects. Secondly, in this sessions, whenever I ask a question, try to answer that how easy it is, how difficult it is. The more we interact, the more we will be available in the class and will remember the data in the classroom itself. My idea will be to make you remember the most of the things in the classroom itself. Now, as we'll proceed, the things will be more clear with us. Now, coming to the first chapter, that is classification of amino acid. See, we know that when it comes to the structure of amino acid, the carbon is having 4 valency. If one of the valency is having the amino group and the acidic group at the same time, amino group, acidic group at the same time, this is referred as amino acid. The third valency is going to be the hydrogen, and the fourth valency can be anything. So I'm writing R. R means it can be any group. It can be hydrogen, it can be carbon, it can be branched carbon chain, it can be sulfur, alcohol, like this. Then there is a concept of alpha amino acid. Now, when we were in the 11th, 12th class, how we used to do the carbon numbering was like this, that we start doing the carbon numbering from the functional group. Here is the functional group. So we say it's carbon number 1, 2, 3, 4, like this. There is one more method of doing carbon numbering. The carbon which is next to the functional group is also referred as the alpha carbon, then beta carbon, then gamma delta, like this. So alpha carbon is the carbon next to functional group. The carbon which is next to the functional group is called as alpha carbon. If we apply this concept on the amino acid, how it is going to be. So here is the functional group, that is the acidic carboxylic acid. Here is the carbon number 1, 2. And if the R is a carbon chain, then it would have been the carbon number 3, 4, 5, like this. Or in other way, I can say that the carbon next to the functional group is the alpha carbon. So here is the alpha carbon. Now here the amino group is attached to the alpha carbon. Amino group is attached to the alpha carbon. So I can say that the amino acid in this particular structure, the amino group is attached to the alpha carbon. So this is referred as alpha amino acid. In fact, in humans, most of the amino acid are found in alpha form. Most of the amino acid are found in alpha form. There are few exceptions, such as beta-alanine. Now, as the name is indicating, beta-alanine means the amino group is attached to the beta carbon. Beta-amino isobutyrate. So the first question for us in biochem will be that which is all of the following are alpha amino acid, except the answer is going to be beta-alanine. There are many others, but this is the common one which they ask. The next is the concept of D and L amino acid. What is D and L amino acid is that when we have the amino acid structure, we try to see that where is the amino group? Is it on the right side or the left side? If it is on the left side, this is called as the L amino acid. If it is on the right side, it's called as D amino acid. The L amino acid, it is found in humans, whereas the D amino acid is found in bacteria. Now, since the bacteria are having the D amino acid, so when we design an antibiotic, we try to kill the D amino acid, not the L amino acid. So one question from pharma is the antibiotics are structured against which amino acid? They are supposed to kill which amino acid? They are supposed to destroy the D amino acid. Now, some more examples that which bacteria are having which amino acid. For example, the Vibrio cholera, it is having D-methionine, D-leucine, D-tyrosine, MLT. The question comes that in cholera, which amino acid are there is MLT. Bacillus subtilis, it is having the D-methionine, D-leucine. So basically, antibiotic, it's all about the D amino acids. But when it comes to one more drug, for example, bleomycin. Bleomycin is a drug which can work against the D amino acid as well as L amino acid both. It can destroy both, means it can destroy the amino acid of humans also. And why we want to kill a human cell in cancers? So that is why the bleomycin, which is used for the testicular cancer, it's a testicular cancer drug, and is going to destroy the D and L amino acid both. The side effect, bleomycin, one important question, it leads to pulmonary fibrosis. Can you tell me some more drugs which leads to pulmonary fibrosis? Pulmonary fibrosis also seen with bleomycin, busulfan. Yes, very good. Amiodarone, nitrofurantoin. Can you tell me one DMRD? Yes, methotrexate. So these are the 5 very important drugs which leads to pulmonary fibrosis. Bleomycin, busulfan, amiodarone, methotrexate, nitrofurantoin. The list is very long. Overall, there are around 40 drugs which are known to cause pulmonary fibrosis. But for the exam perspective, these are the 5 drugs to be remembered. But as of now, the concern was bleomycin that affects the D and L amino acid both. The next is the amino acid. What is the meaning of amino acid? See, the normal structure of amino acid is somewhat like this. The amino group is here and it is free. It can make the bond with the next amino acid. But what if the amino group is not free? If the amino group is bound in a ring structure, likewise, you can see in this example, the amino group is not free, it is bound in a ring structure. If the amino group is not free, it's bound in a ring structure, then this type of amino acid is called as amino acid. So what is amino acid is the amino group, NH2, please correct that. NH2 group is not free, it is bound in a ring structure. This is called as amino acid. Now, the name of this ring is pyrrolidine ring. This is pyrrolidine ring. And the example of this amino acid, it is proline. So this is the second important question. The first important question was, which amino acid is not alpha? That was the beta-alanine. And second is, which of the following is the example of amino acid? Proline. The amino group is not free. It is bound in a ring structure. It's bound in the pyrrolidine ring. Coming to the last basic concept before we really go into the classification part, we are just making some basic terminologies clear with us. The next is the concept of chiral carbon. What is the meaning of chiral carbon? See, the carbon is having 4 valencies. If all the 4 valencies are different, here you can see the A group, here is the B, here is C, here is D. All the 4 valencies are different. If all the 4 valencies are different, then this is called as chiral carbon. And this chiral carbon or the chiral carbon, it is required to make the mirror image. In the 11th class, again, if you remember, we used to make the mirror image, and that is only possible if the molecule contains the chiral carbon. If the molecule does not contain the chiral carbon, we are not going to have the mirror image. So chiral carbon is required to mirror image, to make the mirror image. Now, the problem is that there is one amino acid. The structure of this amino acid, look at this amino acid. Here is the amino group. Here is the acidic group, means it's amino acid. And the third valency is hydrogen. And the fourth valency can be anything. And here it is hydrogen. So the third and fourth valency are same. And if the third and fourth valency are same, I can say it's not a chiral carbon. And this is the structure of glycine amino acid. This is the glycine amino acid. So among all the amino acid, glycine is the only one which does not have the chiral carbon. Glycine does not have the chiral carbon. So it is optically inactive. So the question is very direct, straightforward. Which amino acid is optically inactive or which does not have the chiral carbon? The answer is glycine. So there are 3 questions so far. All are alpha amino acid except beta-alanine. Which of the following is an example of the amino acid? That is proline, and optically inactive is glycine. The same thing is written here. This is kind of a summary for the first 2 pages for us. They all are alpha amino acid except beta-alanine. In humans, we are going to have the L amino acid, whereas the D amino acid, it is found in the bacteria. The example of amino acid, proline, and optically inactive, glycine. Now, once we have clear with the basic terminology part, now we are going into the classification part. But before I start the classification, there is one question that should come to your mind. That Total, we have 22 amino acid, and now I'm saying that we are going to classify them. But the first question that should come is on what basis you want to classify them. For example, if right now there are 22 students are there in the classroom and I tell you that, okay, I want to classify you, then the question is on what basis, right? You will say that, let's say, for example, I can classify on the basis of your current prof, maybe first prof, maybe second prof, maybe third prof, maybe on the basis of your medical school. Some are from different medical schools, some are from different medical school, or maybe on the basis of your year of admission, or maybe year of your 12th or year of birth. So we can have so many bases on which we can classify the things, right? So in the same way, we are having 22 amino acid, and if I want to classify them, on what basis, on what ground? So the first classification that we are going to do is based on the R group. And what is R group? R group is the one that is the fourth valency in the amino acid structure. It can be anything, right? In some amino acid, it will be a hydrogen. In some, it will be a branch carbon chain. In some amino acid, this R will be replaced by sulfur. In some, it will be replaced by alcohol. So we have made the subcategories accordingly to the R group. So the first subcategory is going to be the simple amino acid. What is the meaning of simple amino acid is that this R group is either replaced, this here is the amino acid structure. This R group is either replaced via a simple hydrogen or a simple carbon. If the hydrogen or simple carbon is replacing the R group, they are called as simple amino acid. The structures are, this is the glycine and the alanine. So the mnemonic comes out to be Ga. Ghana is a simple thing to do. Ghana is a simple thing to do. So if they ask that, what are the simple amino acid we have? The answer is Ga. Ghana is a simple thing to do. Ga. And among them, the glycine is the simplest amino acid, because here you can see the structure of glycine. The third and fourth valency are same. It is not having the chiral carbon. It is the simplest amino acid structure. Alanine, it is the most common amino acid or the most abundant amino acid found in the body. The most abundant amino acid that we have in our body is alanine. So this is the simple amino acid GA. The second subcategory is these structures are not important for the exam. They are just to make you understand the things. The second subcategory is going to be branch chain amino acid. Branch chain amino acid means that the R group is replaced by a branch carbon chain. Here you can see that the R group is replaced, instead of R group, we are having a branch carbon chain. If that is the case, these are called as the branch chain amino acids. The example of the branch chain amino acid, leucine, isoleucine, and valine. See how we can remember that. Irrespective of your medical college right now you are in, or maybe India, maybe outside India, or maybe you are intern, whatever it is, you may have seen that, in every batch, there is one person. In your batch also, there was one person who was knowing all the batchmates. All the batchmates were knowing him. He was knowing all the juniors were knowing him, all the seniors, all the professors, and everyone was knowing him. He was having a huge relationship. He was kind of having a huge branch relationship. Now, you and me, we are not having, but that particular student was having. Why? To have a huge branch relationship, the person has to be live-hearted. So, to have a branch relationship, the person has to be live-hearted. Person has to be live-hearted. So the mnemonic is live. To have a branch relationship, the person has to be live. That is leucine, isoleucine, and valine. Leucine, isoleucine, and valine. Now coming to the third subcategory, that is the alcohol-containing amino acids, your favorite one. Alcohol-containing amino acid or the hydroxyl group containing amino acid. The example of them is serine and threonine. But before I go to that example, I want to share one incident with you. No need to write this part, just see here everywhere. What happens is in the North Asian countries, North Asian country, be it Russia, China, or maybe the Central Asian countries, the Kyrgyzstan, Kazakhstan. Now, there is a tradition that whenever a guest will come to their house, they are going to offer alcohol. In India, unfortunately, we don't do that, right? Whenever a guest comes to us, we don't do the this let okay, taru piyo, right? We don't do that. But in these countries, it's a tradition. Now, since I was traveling in these countries very extensively to take the classes there, so I was knowing that tradition. One day, what happened was, few students from China, They have called us that, sir, we want to have your class there in our university. So we said, okay, fine. And the date and time was decided. This was the city of Nanjing where the class was supposed to happen. So I landed at Shanghai airport around 5 p.m. And there were 2, 3 students has came to take me from Shanghai to Nanjing. It is a 3 hours distance. And when we were meeting for the first time, it was way back around 8 years back, 2017, 2016, 2017. So now what happened was we started talking very casually, and one of the students told me that, sir, from tomorrow onwards, we are going to have a very hectic class. And on the last day, from the class itself, you have to travel back to the airport. So on the last day, we are not going to get the time. So he said to me, sir, let's have a party today. We are not going to have the time on the last day. I understood their meaning directly, so I bluntly said that no, I don't drink. And there was a killing silence in the car for the few minutes. Then again, I started talking casually and make the environment little more casual again. By 8 p.m., we reached Nanjing, and they were supposed to drop me at the hotel. When they dropped me at the hotel, now we were more familiar with each other. So one of the students again told me that, sir, we understood that you don't drink. But what you do is that you just come and sit. We just need a reason to start. Baki hum dekh lenge. So I said, okay, fine. They were insisting so much. So I reached there at 9 p.m. The time and place was decided. I reached there 9 p.m. And now what they did was there was already 7, 8 students who were sitting there, and I was sitting here. And they have given me a tetra pack of juice, and they said, sir, as and when you want, you take it. And they started drinking, and we were talking casually again. Then around maybe after 1 hour, they were little high, and one of the students told me, sir, uh, you never know when the life is going to end, and we don't know each other. Maybe we will not meet again nowhere. So why don't you also try at least once in your life? So I said, okay, fine, give me one. So I took the first one, and I liked it. And actually, everyone likes that. Then I asked on my own for the second one. Then I asked for the third one. As soon as I asked for the fourth one, the student who was organizing the class, who has collected the money from the other students, he was thinking that this guy is drinking for the first time. And he's thinking that I am not aware about hangover and all this stuff. So he was thinking that how I am supposed to take next day 10-hour class. It is going to be difficult for me. And if the class doesn't happen, all the students will blame this particular guy that he has collected the money, right? So he has to stop me. And since we all were from North India, so we were talking in Hindi only. So what he said to me was, sir, please stop. This is alcohol. In Hindi, what he said was, sir, he said to me, sir, Thero, this is alcohol. Sir, Thero, this is alcohol. So the alcohol containing amino acid, it is Sir Thero. Sir Thero, this is alcohol. Now, when it comes to this incident, there is no such incident happened. This is just to make you remember the things. Sir Thero, this is alcohol. But there is one more amino acid that is there, which contains alcohol. And but deliberately we are writing that in the separate line, and that is tyrosine. Tyrosine also contains hydroxyl group. But why we are writing in a separate line? In the next 5 minutes, we'll complete that and we'll come back here and we'll write down that why we have written tyrosine in the separate line. Coming to the fourth subcategory of the amino acid, the sulfur containing amino acid. The example is methionine and cysteine. How we can remember is what is there inside the meaty cyst. Inside the meaty cyst, what is there? Sulfur. Meaty cyst contains what? Sulfur. Meaty cyst, sulfur. The fifth subcategory is the aromatic amino acid. As the name is suggesting, the amino acid which is having a particular type of smell is called as aromatic amino acid. Now, to understand this, let's do one experiment. What we do is, for example, we are sitting in a closed environment, let's say a big hall, which is completely closed. No, nothing is there. No windows, nothing is there. It's closed. And we do experiment there. We bring some tire there and we burn that tire in that room. After some time, there will be a smell of that tire in that room everywhere. And if you want to get rid of the smell, you need to turn on the exhaust fan. If we turn on the exhaust fan, then the smell will go. So in other words, I can say that wherever there is smell, If you want to get rid of the smell, you have to turn on the fans. You have to turn on the exhaust fan. So we are discussing the aromatic amino acid which are having a smell, and wherever there is smell, the fan has to be there. So whichever amino acid will have the smell, they will have fan in the name. So can you tell me some amino acid which are having fan in the name? Which amino acid is having fan in the name? For example, tryptophan, fan in the name. So it is aromatic. One more we can say phenylalanine, fan in the name. Phenylalanine, tryptophan, phenylalanine. Now, what was the smell? The smell was of tire. The smell is of tire. So I can say one more aromatic amino acid, that is tyrosine. So tryptophan, phenylalanine, and tyrosine. There is one more, histidine. Now, histidine is not fitting in the mnemonic, so we'll put some hard work to remember that. But otherwise, the aromatic amino acid are tryptophan, phenylalanine, tyrosine, and histidine. And these aromatic amino acid has one more property. They absorbs the UV rays very nicely. If you put them in the sunlight, they are going to absorb the UV rays. The frequency of the UV rays is 250 to 290 nanometer, and they absorb it very nicely, particularly the tryptophan. And this has been asked in your exam. The maximum UV absorption is seen with which amino acid? It is seen with tryptophan. Among all the aromatic amino acid, the maximum UV absorption is tryptophan. If they want to make a difficult question, instead of UV rays, they will use the word that which amino acid have more potency to absorb the rays which is between 250 to 290 nanometer, because this wavelength is for UV rays and the maximum is for tryptophan. The next subcategory is acidic amino acid. See, how we write the acid is like this. We write the acid like COOH, or we write like COO negative. COH or CO negative. So I can say that on the acid, what charge we are having is the negative charge. Acidic amino acid are the amino acid which have excess amount of acid, and acid have negative charge. So I can say the acidic amino acid must have negative charge on them. They have the negative charge. The examples are aspartic acid, glutamic acid. But if they want to ask you this question that which of the following is a acidic amino acid, definitely they will not give you the option aspartic acid, glutamic acid. They will use the other name. The other name of aspartic acid, aspartate. The glutamic acid is glutamate. So, like it is like the same. For example, lactic acid. What is the other name of lactic acid that we are using is lactate. Pyruvic acid, the other name is pyruvate. In the same way, the aspartic acid, glutamic acid, we use the alternate name, the aspartate and glutamate. These are the acidic amino acid. The next is the amide group containing amino acid. The amide, but before we go to amide group containing amino acid, we should know that what is the meaning of amide is. What is the meaning of amide is? Because when I said sulfur containing amino acid, you understood sulfur means S. When I said alcohol containing, you understood alcohol means OH. But what is amide? Amide means CONH2. So basically, we are searching for the amino acid which is having this CONH2 group on that. And here is the catch that amide loves the acid. Wherever is the acid, the amide will go and will attach on that. And just now we have discussed there are two amino acids which have excess amount of acid on them. One is the aspartate and second is glutamate. They have excess amount of acid and amide loves that. So amide will go and will attach on aspartate. If it attaches on aspartate, what we are going to make? Aspargine. Aspargine. The amide can go and can attach on glutamate, and we are going to make glutamine. So aspargine and glutamine, these are the amide group containing amino acid. Now, here are two questions. The most abundant amino acid in the body and plasma. I already told you the answer for the first one. The most common amino acid in the body, it is alanine. But if they particularly ask in the blood, in the plasma, which is the most common, then the answer is glutamine. Glutamine, right? Now, the last subcategory on the basis of R group, the basic amino acid. Because the acid was having negative charge, the basic will have the positive charge. These are the positively charged amino acid. And the example of the positively charged amino acid, the basic amino acid, histidine, lysine, and arginine. How you will remember this? See, if you, when you will be posted in the oncology ICU, you will see that there are some patients who are in terminal stage. We can't do anything. The cancer is already widespread. They are going to die. The patient also knows that. The family member also knows that. We also knowing that. So by chance, for example, obviously we'll not do this, but by chance, if we do like this, if there is a person who knows that he's going to die because of widespread cancer, and if you ask that, sir, what is your last urge in your life? What is your last wish that you have? The last urges are going to be very basic. They are going to be very simple, right? So he will say that I want to meet that particular person. I want to visit that holy place. Something like this will be there. So when it comes to the basic amino acid, the mnemonic will be his last urge is very basic. His last urge is very basic. Histidine, lysine, arginine. His last urge, very basic. Among them, the most basic amino acid is arginine. Among them, the most basic is arginine. Right? So this is all about the first classification that is based on the R group. Now coming to here is one information is left, the glutamate and glycine. See, the glutamate in the physiology we will discuss that it works as an excitatory neurotransmitter, whereas the glycine works as an inhibitory neurotransmitter. Excitatory and inhibitory. Now coming to, this is the first classification that is based on the R group, where we have discussed the simple amino acid, the branch chain amino acid, the sulfur containing, alcohol containing, acidic, basic, amide group containing amino acid. Now coming to the second classification that is based on water solubility. It is going to be a very easy classification because out of 22 amino acid, there will be some which are water soluble, there are some which are water insoluble. But we are not going to remember like a single list because it is going to be difficult for us if we remember like a single list. Instead of that, we'll remember the families and that will make the things easier for us. See how. For example, if I ask you that, or I want, first I am sharing one fact that anything which is having a charge, these things are going to be water soluble. Anything which have a charge, either positive or negative, if the charge is there, that is going to be water soluble. And you know there are two families, two amino acid families which have the charge. There are some amino acids which have the negative charge and some have the positive charge. And what are these families? The negatively charged amino acid are the acidic amino acid, and the positively charged is going to be the basic amino acid. And we know that if they have anything is having a charge, they are going to be water soluble. So they are going to be, these two families are water soluble. And who are the family members? In the acidic amino acid, we have the aspartate and the glutamate. Whereas in the basic, the mnemonic was his last urge is very basic. So these are the basic amino acid. Then there is one family which loves the acid, right? And what is the name of that molecule which loves the acid? The name of that molecule was amide. So I can say that wherever is the amide will go means the acid is there. And if the acid is there, that is going to be water soluble. So the amide group containing amino acid, they are going to be water soluble. And what are the examples? They are going to be the asparagine and the glutamine. Glutamine. Then one more thing. You may have confusion about the charge. You may have the confusion about amide, whether they are water soluble or not. But there is no doubt that alcohol is water soluble, right? We are every day we are taking few ml of vodka, whiskey, and we are diluting in water or Coca-Cola or something like that, right? So alcohol is going to be water soluble. So the alcohol containing amino acid, they are going to be water soluble. And you know what is the mnemonic for alcohol containing amino acid? Sir, thero. This is alcohol. So they are going to be water soluble. So now we know four families which are water soluble: acid, base, amide, and alcohol. They are going to be water soluble. But if you're listening very carefully, can you answer me that in the alcohol family, there was one more amino acid was there? What was that amino acid which we have written in a separate line in alcohol family? Sir, but there was one more. Yes, right. They said Shakshi, it was tyrosine. Now, why we have written tyrosine in a separate line? See, the tyrosine, it contains alcohol. Tyrosine, it contains alcohol. Despite it contains alcohol, it is not very good water soluble. It's not very good water soluble. So that is why we have written tyrosine in the separate line in the category number 3. So let's complete that statement also. Let's go to the category number 3, where we have written the alcohol containing amino acid and we have written tyrosine in the separate line. Tyrosine also contains the hydroxyl group, but it is not water soluble. It's not very good water soluble molecule. It is more of water insoluble. It's not water soluble. So now we understand that if they ask that which of the amino acids are water soluble, you will say there are four families which are water soluble: acid, base, amide, and alcohol. So here it is written. The alcohol containing, the water solubility, the water soluble amino acids are also referred as the hydrophilic amino acid, also called as the polar amino acid. And there are four families in that: the acidic amino acid, the basic amino acid, the amide group containing, and the alcohol containing. And we know the family members also. Plus, there are two individual amino acid. They are also water soluble. Their entire family is not water soluble, but individually they are water soluble. And what are these two amino acid? That is the glycine and cysteine. See, glycine belongs to simple amino acid and cysteine belongs to sulfur containing amino acid, but their entire family is not water soluble. These two are the individually water soluble, glycine and cysteine. So there are total four families and two individuals which are water soluble: acid, base, amide, alcohol, glycine and cysteine. So here you can see that if they ask that which are the water soluble amino acid or the polar amino acid, we can divide into two categories: charged and uncharged. In the charged, there are two families will come: the acidic amino acid and the basic amino acid. Whereas the uncharged, they are going to be amide containing amino acid, alcohol containing amino acid, and there are two individuals which are there: glycine and cysteine. Glycine and cysteine. Now, remaining all are going to be water insoluble, means hydrophobic or non-polar. So we will just remember on one side, the other side, the water insoluble, remaining all are water insoluble. But if they ask that among all which are remain water insoluble, which is most water insoluble? The most water insoluble is going to be isoleucine. Isoleucine is the most water insoluble. And in the same way, if they ask that which is the most water soluble, the answer is arginine. Arginine is the most water soluble. So the most water insoluble isoleucine and most water soluble is going to be arginine. But if they want to ask you a difficult question, they are going to create a spectrum and then they will ask the question. What is the meaning of that? For example, if I give you 22 amino acids and I tell you that, okay, arrange them in according to their ranks. The most water soluble, rank 1, then the rank 2, then the rank 3, and the most water insoluble, rank 22. If you arrange in that manner, it will be like this. The most water soluble is going to be arginine. Then there will be some, then the last thing that will come is isoleucine. If you look at the tyrosine, it is coming in the gray area. Neither it is completely water soluble, nor it is completely water insoluble. It is somewhere in the gray area. But if you have to select tyrosine as one, then yes, definitely we will select as more as water insoluble. More as water insoluble. It is somewhere in the gray area, but is considered as water insoluble. But how it is going to help us in solving the MCQ? Now, look at these three MCQ and try to find the best answer. They are asking more or less the same thing, but the options are changing. The first question, which is the water insoluble amino acid. So whenever such questions will come, you will always search for the family. Sir, Thero, it belongs to alcohol, and we know that alcohol is going to be water soluble. They are water soluble. Then we are left with isoleucine and tyrosine. Among them, if you have to select that, which is the best answer, which is the water insoluble amino acid, then what you are going to select? Isoleucine. Although both are water insoluble, but the best answer is going to be isoleucine. In the same way, the second one, the identify the water insoluble amino acid. The options are serine, threonine. They are alcohol containing amino acid. Sir, Thero. Then we have the acid. Acid contains negative charge, and negative charge means it's water soluble. And then we have the tyrosine. Here we will consider tyrosine as water insoluble. So if you see the first question and second question, in first one you have considered tyrosine as water soluble, whereas in the second one you have considered it water insoluble, depending on the other choices. In the same way, the last question, which is the least hydrophilic? The least hydrophilic, again, here you can see Sir, Taro, they are basically alcohol containing amino acid, aspartate is acid, and tyrosine. So again, the answer is tyrosine, right? So if they want a difficult question, you need to solve like this according to the family. Then you will decide that whether the tyrosine will belong to water soluble or insoluble, depending on the other choices. Now, the third classification that is based on the metabolism part. we are having 22 amino acids. And if we do the metabolism, there will be some amino acid after the metabolism, they are going to generate the ketone body. If they generate the ketone body, they are called as ketogenic amino acid. There are some which will convert into glucose after the metabolism. They are called as glucogenic amino acid. And there are some which can convert into ketone body as well as glucose both. They are going to refer as amphibolic. They can convert in any of the one, depending on the requirement of the body. So we can say that based on the metabolism, the amino acid is divided into three categories: ketogenic amino acid, glucogenic amino acid, and amphibolic amino acids. What are the ketogenic amino acids? The ketogenic amino acids are going to be leucine and lysine. Now, but before I go to that, you answer me one question. No need to write this. I'm writing some options also for you. First, let me write the option and then you will select the answer. My question is, which is the most common pet animal in India? Which is the most common pet animal in India? What do you say? B or D? In India, which is the most common? See, definitely cat is not that common, right? In India, we don't have cat so commonly as a pet animal. Yes, some of you are saying dog and some of you are saying cow. Now, let me add one more option for you. Buffalo. Is it changing your answer now? Yes, it is neither the cow or buffalo because they are not the pet animal. They are not sitting with you in the sofa in your drawing room. The cow or buffalo, they are not sleeping with you in your bedroom, right? In your bed. So in India, the most common is going to be dog. Okay. But let's change the question then. If I ask you that, which is the most common pet animal worldwide, then the answer will be cat. And in cat, just consider, let's consider, maybe I'm right, maybe wrong, but let's consider cat is most common pet animal worldwide. If I ask you that, what is the most common name of a pet cat worldwide? Cat is the most common. What is the most common name of a pet cat? It is not what you are thinking. That is not the correct one. The correct one is Lucy. Lucy is the most common name of a pet cat. How it is helping us? How it is going to help us? See, when it comes to ketogenic amino acid, we know that amino acid we are divided into three categories: ketogenic, glucogenic, and amphibolic. When it comes to ketogenic, that is cat, ketogenic cat, the most ketogenic is going to be leucine, leucine. So the ketogenic is leucine and lysine. Leucine and lysine. Cat is leucine. The most ketogenic, leucine. So here also it is written the most ketogenic is going to be leucine. Leucine is the most ketogenic. When it comes to the amphibolic, in amphibolic family, they are going to be in amphibolic family, there are going to be the four amino acids. And what are these four amino acids will be like this: tryptophan, phenylalanine, tyrosine. Can you tell me that it is looking very similar like which family? Amphibolic is very similar to which family? Fan in the name, tire. It is very similar to which family? Yes, rightly said, Apurva. It is very similar to aromatic family, right? So I can say that amphibolic, it's easy to remember. We'll say that amphibolic is basically aromatic. But there is one change. What was the fourth one? The fourth one in aromatic family, who was the fourth one? Histidine. But here it is going to be isoleucine. So that is one change that is there. And remaining all are going to be the glucogenic. So if they ask that, based on metabolism, we are having three families, the ketogenic, the amphibolic, and glucogenic. We'll remember the first two. Ketogenic is going to be leucine and lysine. Amphibolic is similar to aromatic, and glucogenic is remaining all. And if they ask that, which is the most ketogenic is going to be the most cat is going to be the leucine, whereas the most glucogenic is going to be alanine. I will ask this question again tomorrow about the most glucogenic amino acid, alanine. When we'll do the gluconeogenesis, I will ask that which is the most glucogenic amino acid. It is going to be the alanine, right? The most water soluble is arginine, Prashant. Now coming to the fourth classification, the last classification based on the nutritional requirement. Now we are having the 22 amino acid. Based on the nutritional requirement, The amino acids are divided into three categories: essential, semi-essential, non-essential. What is the meaning of that? We have heard about these words even in the schools, right? Essential amino acid means the amino acid which you must take in your diet. You can't synthesize them. Non-essential is the amino acid, even if you don't eat on your own, the body will make it. So you don't have to worry about non-essential. They will take care of themselves on their own. They are non-essential. The body will make it. You don't have to eat them in the diet. Semi-essential is the amino acid which you have to supplement in the diet when you are in the growing age, means in the pediatric age group. After that, you don't have to supplement. Means the semi-essential is the one which we are making, but not in the adequate amount. So essential amino acid we are making and you don't have to eat, you will make in adequate amount. Semi-essential you are making but not in adequate amount, so you have to eat at least some amount. And non-essential you don't have to worry. You are making it and you don't have to eat it. Your body will take care of them easily. Now, when it comes to the essential amino acid, out of 22, we need to remember that there are total 9 amino acids which are essential. How to remember that? We are going to use a mnemonic TV till 9 p.m. H. TV till 9 p.m. H. These are the essential amino acids. TV till 9 p.m. H. Now, what is TV till 9 p.m. H? The full form is written here. TV till 9 p.m. H. There are total 9 amino acids which are essential. When it comes to semi-essential, there is one that is arginine. And remaining all are going to be non-essential, means you can make them in their body. But now there is a problem. The problem is that key TV till 9 p.m. H, you may have seen that in some books, histidine is written as semi-essential also in some books. But now I'm writing in essential category. This is one problem with this mnemonic. And second, the major problem with this mnemonic is if they ask that all are essential amino acid except, you will start using the mnemonic TV till 9 p.m. H. But if you see that there are two T's in the mnemonic, one T is tryptophan, second T is threonine. But can you tell me one amino acid which starts with the T? Apart from tryptophan and threonine, there is one more amino acid which starts with the T, tyrosine, right? So tyrosine, you have to remember that tyrosine is non-essential amino acid. Means when you use this mnemonic, how you have to use a mnemonic? If they say that all of the following are essential amino acid, what is the mnemonic? TV till 9 p.m. is essential and tyrosine is non-essential. Then it is complete mnemonic. Otherwise, they will confuse you on T. Which T is essential, which is non-essential. So tyrosine is non-essential. So tryptophan threonine is essential, tyrosine is non-essential. Now coming to the semi-essential is going to be the arginine and the remaining is going to be the non-essential. This histidine, some books consider histidine as semi-essential also. So now it is going to be a controversial question and then we have to see the other option and based on that we need to decide. See how we will decide. Look at these questions and you give me the answers. Identify the non-essential amino acid. Non-essential amino acid. The options are TV till 9 p.m. H. So here it is TV till 9 p.m. H. And we know that tyrosine is non-essential. Starts with the T, but it's non-essential. Now, the next is, which is not essential amino acid? Again, let's use the mnemonic TV till 9 p.m. H. So now the answer is going to be arginine. It is semi-essential. It is not essential. Here we are considering histidine as essential amino acid. Here we are considering histidine as essential. But look at the next question, which is not essential. TV till 9 p.m. H. Now you can see that all are coming from the mnemonic and they're asking which is not essential. Then what will be the answer? The answer is going to be histidine in that case. In that case, we'll see that, okay, the examiner might be considering histidine as semi-essential because other are clear-cut essential. So histidine is some of in the graysy area. It may be considered as essential, maybe semi-essential, but most book consider as essential. So if nothing is coming, nothing is getting into fit, then we'll consider histidine as essential, TV till 9 p.m. H. Earlier, we were just having 20 amino acids. The 21st and 22nd, they were recently added. So we should know what is the name of them, what is the codon for them. Just few details about them. 21st amino acid recently added. The name of that, selenocysteine. The 22nd is pyrrolysine. These are the ones which are recently added. Now, what is the importance of that? See, if, like you are having the roll numbers in your exam, likewise in the same way, every amino acid have a codon, like their roll number, identification code. The selenocysteine, the code is going to be UGA, and the pyrrolysine is UAG. We will discuss about these codons in more detail when we will come to the chapter of DNA and RNA. At that time, we will discuss in more detail. But as of now, every amino acid have their kind of roll number. They are called as codon. We can't remember all of them, but we are discussing about these two because they are recently discovered. So we should know the name and the codon. Okay, now before we go there, I have 2, 3 questions for you. If you go to the rural part in the northern India, in the in the in the rural part of northern India, what happens is that uh, if there is a newly married couple and husband is sitting with some elderly people and the wife wants to call the husband, the wife will not call the husband by his name in the rural part of northern India, right? They will use some alternate, maybe maybe AG, OG, or maybe with the name of their child. But since they are recently married, so they don't have any child. So that option is eliminated. Now, how this wife is going to call then? Maybe AG, maybe OG, something like this, right? Because they are recently married, so there is still pyar is there. And wherever there is pyar, there is AG, OG, Sunoji, something like that is there. Wherever there is pyar, there is going to be AGOG. Wherever there is pyar, there is going to be AGOG. So pyrrolysine, pyar, it is UAG. One more point is, if I ask you one more question, that selenocysteine, what do you think that selenocysteine is made up of what? What do you say, selenium or cysteine? Who is the precursor amino acid of selenocysteine? What do you say, selenium, cysteine, or both? Yes, the answer is none. It is made up of what? It is made up of, it is the precursor amino acid is serine. It is made from serine. From serine, we make selenocysteine by doing some co-translational modification. In the same way, how the pyrrolysine is made? The pyrrolysine is made is you take the lysine and you attach the pyrrole ring. On the lysine, if you attach the pyrrole ring, it's called as pyrrolysine. Here you can see the structure. Look at the structure of lysine and pyrrolysine. The additional thing that you can see in the structure is the ring. These structures are not important. That is just to make you understand that in the pyrrolysine, this is the additional thing which you are having. This is the additional thing. This is the ring which is there. The name of this ring is the pyrrole ring. So if we attach the pyrrole ring on the lysine, it is going to be the pyrrolysine. This is all about the first chapter, that is the classification of amino acid. With every chapter, we will do 3 times revision. First, when we are discussing the things, you write down the things, that is kind of a revision. The second, at the end of the chapter. And then we will write down the summary also. That is going to be the third revision. And in fact, towards the end of the class, we will try to revise the entire day very quickly in 5 to 10 minutes. So that is again going to be a revision for us. So you just be with me and I will make sure that you will remember most of the things. But for that, you need to continuously answer out the things, what is being asked. So you answer down the things. We have discussed the classification. That is, first we have discussed based on the R group. You just tell me the mnemonic. Which is the simple amino acid? The simple amino acid, what is the mnemonic? Ga. Gana is a simple thing. Glycine and alanine. Then we have discussed the branch chain amino acid. To have a branch relationship, the person has to be live-hearted. Leucine, isoleucine, valine. Then the alcohol containing amino acid. What are the alcohol containing amino acids? Sir, thero. This is alcohol, but there is one more, tyrosine. Then is the aromatic amino acid. The aromatic will have the smell, means the fan in the name, such as tryptophan, phenylalanine. What was the smell? Tyrosine. And can you tell me that what was the fourth one, which was not there in the mnemonic? Yes, very good, histidine. Then we have discussed the acidic amino acid. What is the charge on acidic amino acid? Negative charge. And what are the example? Aspartate and glutamate. Who loves the acid? Which family loves the acid? Amide. So the amide group containing example, asparagine and glutamine. The basic amino acid. How to remember the basic amino acid? His last urge is very basic. Histidine, lysine, arginine. His last urge is very basic. So these are the various. And one more we have left for sulfur containing amino acid. What is sulfur containing? Meaty cyst. What is there inside the meaty cyst? Sulfur. Then the second classification that we have discussed on the water solubility. Can you tell me four families which are water soluble? The four families which are water soluble: acid, base, amide, alcohol. And can you tell me two individuals which are water soluble? Two individuals which are water soluble, not the entire family, but the individuals: glycine and cysteine. Remaining are water insoluble. The third classification we have discussed is based on the metabolism: ketogenic, amphibolic, and glucogenic. I want you to remember the most ketogenic. What is the most common name of a pet cat? What is the most common name of pet cat, Rahul? It is leucine. Leucine. Leucine and lysine. Leucine is the most ketogenic. When it comes to amphibolic, it is similar to aromatic. And the the glucogenic, it is the most glucogenic is going to be alanine. Lastly, we have discussed the classification based on the nutritional requirement: the essential, semi-essential, non-essential. What is the mnemonic for essential? TV till 9 p.m. H. The only semi-essential arginine, and remaining all are non-essential, right? So this is all about the first chapter, that is classification of amino acid. Now there are four questions for you. The first few questions you will write down on your own. These are the previous year questions which has been asked in different exams. That is kind of a homework for you that you will write down the answers afterwards once we'll complete the class. But right now I want these four answers from your side. Write down these four answers. Basically, you have to find the common family members between the two families. There is an amino acid which is aromatic as well as having the hydroxyl group. Having the hydroxyl group as well as aromatic. Yes, very good, Aditya. Hydroxyl group, Sir, Thero, and Tyrosine. Among them, which is aromatic? Sir, Thero, and Tyrosine. Among them, which is aromatic? Tyrosine. The next one, aromatic and basic. What are the basic? His last urge, histidine, lysine, arginine, is very basic. His last urge. And among them, which is aromatic also, his last urge, among them, who is the aromatic? Histidine. The next is the simple amino acid, but water insoluble. What are the simple amino acid? Ga, glycine, and alanine. Among them, which is water insoluble, not the water soluble. Yes, the answer is alanine, because glycine is water soluble. Charged, positively charged, and polar. Yes, definitely. Anything which is having charge is going to be polar only. The positively charged and water soluble, they are going to be what? The acidic amino acids. Sorry, the basic amino acid. Positively charged, the basic amino acid, his last urge. His last urge. That is going to be the positively charged and polar, right? So this is how they will ask the question, and sometimes two families, they will try to find which is the common one. The remaining are the easy questions. You can solve them on your own. Now coming to the next concept, that is the biosynthesis of urea. Now, the next 5 to 7 minutes are very important for us to understand this overall topic of amino acid metabolism, because once we'll understand this, then the things will be easy for us. Now, what is the meaning of this chapter? Biosynthesis of urea. We understand that we are synthesizing urea in our body, but the question is why? Why you want to make urea in your body? See, whatever protein we are eating, proteins are made up of amino acids. So when we break the protein, we make amino acid. When we break the amino acid further, we make ammonia. And the problem is, ammonia is a very bad molecule. It's a very toxic molecule. We don't want ammonia. So we want to convert into a non-toxic form so that we can throw it out. And the non-toxic form is urea. So we need to convert the ammonia into urea, and this conversion can take place only in one organ, and that is the liver. The liver has the enzyme to convert the ammonia into urea. The urea is non-toxic, and then this urea will come in the blood. When the urea molecules comes in the blood, that is called as the blood urea nitrogen. And this blood urea nitrogen will reach to the kidney and will be filtered out and will be thrown out. So now we have understood 2 points here, that why we want to make the urea and where we are doing that. The urea synthesis, why we are doing is to convert a toxic molecule ammonia to convert into non-toxic form, that is urea. And where it is happening, only one place, that is liver. But now the question is how? How we will convert the ammonia into urea? See, one point is very clear. If you want to convert the ammonia into urea, from wherever in your body you have ammonia, you have to send it to the liver so that the liver can convert into urea. But how we will send it? The problem is the transport of ammonia is very difficult because it's a toxic molecule, right? We can't transport it directly. So the body tissues has to find the alternate to send the ammonia molecules to the liver. How? I will divide the body tissue into two groups. The liver is the hepatic tissue, and other than that, the muscles and all, the heart, the muscle, the brain, they are extrahepatic tissue. Now, in the extrahepatic tissue, we are having the different proteins. Let's consider one. For example, in my bicep, in my muscle, there is one protein, and it is made up of different amino acid. If I break this protein in the extrahepatic tissue, I will get different amino acids: glycine, valine, cysteine, glutamine, like this. Now, what happens is, means in my muscles, we are having the different amino acid. If I break them further, I will make ammonia. But I don't want this ammonia in the muscle. I want this ammonia in the liver because liver has the capacity to convert the ammonia into urea. So what this tissue will do is the tissue will request all the amino acids. For example, the muscle. The muscle will request all the amino acid that please go to the liver and donate your ammonia in the liver so that the liver can convert ammonia into urea. But none of the amino acid was getting agreed to go to the liver and donate the ammonia. No amino acid was getting agreed. Then out of 22 amino acid, one amino acid got agreed, and the name of that was glutamate. The glutamate said that, okay, I will go to the liver and I will donate my ammonia there. But the rest of the amino acid, they were not agreeing. So what the muscle will do, what the extrahepatic tissue will do, because the glutamate was the amino acid who was agreeing to go to the liver. So the extrahepatic tissue, whatever amino acid is there in the extrahepatic tissue, whether it is alanine, aspartate, valine, whatever it is, the extrahepatic tissue will try to convert those amino acid into glutamate, because the glutamate is ready to go to the liver and donate the ammonia. So the extrahepatic tissue first converts all the different amino acid into glutamate. And that process is called as transamination. And once you do this, now these glutamate molecules will come in the blood. Via the blood, these glutamate molecules will travel and will reach all the way to the liver. So now the problem is solved because the glutamate is the one who agreed to give the amino to the ammonia to the liver. So we converted all the different amino acid who was not agreeing. We will convert them into glutamate. The glutamate will go to the liver and will donate the ammonia. And if you remove the ammonia from the glutamate, that is called as deamination. And this process occurs in the presence of oxygen. So it's called as oxidative deamination. And this is what we wanted, right? We wanted the ammonia to reach to the liver. So now you know the two steps, how you load your liver with ammonia. First, you convert all the amino acid into which amino acid? Glutamate. And that process is called as what? Transamination. Then this glutamate will go to the liver, and in the liver, we will take out the ammonia from the glutamate. That is called as oxidative deamination. When these things were happening, there are three more amino acids. They came forward and they said that, okay, please don't convert us into glutamate. We will also go to the liver and we will donate our ammonia there. And what were these three amino acids? The mnemonic for that is not IPL, it is TPL, threonine, proline, lysine. These three amino acids, they said that please don't do transamination for us. Don't convert us into glutamate. We will also go to the liver and we will undergo oxidative deamination directly and will donate our ammonia directly there. So these three amino acids will not undergo transamination. They will also go directly. So technically, there are four amino acids which does not undergo transamination. What is transamination? You convert the different amino acid into glutamate. And there are four which will not undergo transamination. One is threonine, proline, and lysine. What is the fourth one? Can you tell me that? What is the fourth amino acid which will not undergo transamination? Means which amino acid will not be converted in the extrahepatic tissue? Yes, the glutamate itself. The glutamate amino acid, what is there in the proteins, they will not be converted because they are already glutamate. So technically, there are four amino acids which we don't have to do transamination, right? And now we have got the ammonia in the liver. But now there is one problem. Sometimes there are few ammonia molecules which are freely produced in the extrahepatic tissue, maybe the muscles, maybe the brain. We have some free ammonia molecules. And we know that the conversion can only happen in the liver. So we need to transport these ammonia molecules to the liver. But the problem is these ammonia molecules are very toxic. How to transport them from the muscles, from the brain? How to transport them? We require the transporter from the muscle. Who is going to bring the ammonia? The name of the transporter, alanine. And who is going to bring it from the brain and the other tissue? Glutamine. These are the transporters. They will take these ammonia molecule and they are going to transport these ammonia molecule all the way to the liver. And now the ammonia has came to the liver. So actually there are four molecules by which you are getting the ammonia in the liver. Can you tell me four methods by which you are getting the ammonia? Can you tell me four amino acids by which you are getting the ammonia in your liver? One is glutamate. Very good. Second is TPL, threonine proline lysine. The third is alanine. The fourth is glutamine, right? These are the four things which are somehow bringing the ammonia to your liver. The glutamate, the TPL, the alanine, and glutamine. Now, once your liver is loaded with the ammonia, this was the step number 3, the transporters of the ammonia. So the step number 1 is the transamination. Step number 2 is oxidative deamination. Step number 3 is the transporters of ammonia. Now the liver is loaded with ammonia. What the liver will do with this ammonia? Liver will convert this ammonia into urea, and that is called as what? The urea cycle. The liver will convert the ammonia into urea, that is urea cycle. These urea molecule then will come in the blood. When the urea comes in the blood, it is referred as the blood urea nitrogen. And these blood urea nitrogen will reach all the way to the kidney and will be filtered out. And this is how we will eliminate the urea from our body. So there are 4 steps in the biosynthesis of urea. What are the 4 steps? The first, you convert all the amino acid into glutamate, that is transamination. Second, this glutamate will go to the liver and you will take out the ammonia. It's called as oxidative deamination. The third is the transporters such as alanine and glutamine. And the fourth is going to be the urea cycle where you convert the ammonia into urea. Now we will see the details of each of the steps, but once you have understood the basic idea, you are going to give the answers for the next of the all the 4 reactions, because we have got some basic idea out of this diagram. So the first, the transamination. Can you tell me that transamination occurs where? Hepatic tissue or extrahepatic tissue? The transamination is occurring where? The transamination is occurring in the extrahepatic tissue. What we are doing in transamination, we are converting the different amino acid. Can you tell me that? The different amino acid you will convert into which amino acid? When you do the transamination, we convert all the different amino acid into glutamate. Convert into glutamate. For that, the name of the enzyme is transaminase. We are doing transamination, so the name of enzyme is transaminase. And for the coenzyme is going to be PLP. It is the active form of vitamin B6. The full form is written below. PLP, pyridoxal phosphate. The full form is here. Everything, the full form will come. So PLP, it is the active form of vitamin B6, pyridoxal phosphate. This is how we do this. Now, can you tell me that what are the 3 more amino acid which said that please don't do transamination, will go directly? What were the 3 more amino acid which agreed to go directly? That was TPL. So there are 4 amino acids which don't undergo transamination, right? One is the glutamate and then the TPL, threonine proline lysine. They don't undergo transamination, but rest of the amino acid, they undergo transamination, right? The aspartate, the alanine, they undergo transamination. Glycine, the valine, they are undergoing the transamination. But how the aspartate will convert into glutamate, how the alanine will convert into glutamate. There are so many transamination examples will be there. We can't remember all the transamination examples. So we are just going to remember 2 examples of transamination. 2 examples. How aspartate converts into glutamate, how the aspartate converts into glutamate, and how alanine converts into glutamate. These are the 2 examples we'll try to remember of transamination. And why? Because they are clinically very important. How the aspartate converts into glutamate. See, when this reaction happens, transamination, the product is glutamate, that is fixed. But what is the byproduct? Here the byproduct is oxaloacetate. When you do the transamination of aspartate we get the glutamate, along with that oxaloacetate. When you do the transamination of alanine, we get glutamate, along with that we'll get pyruvate. Now, why these reactions are clinically important? The name of the enzyme is going to be transaminase, but if I tell you the specific name of enzyme for the transamination of aspartate, the specific name of enzyme will be like this: serum glutamate, G for glutamate, O for oxaloacetate, transaminase. Serum glutamate oxaloacetate transaminase. In the same way, when you do the transamination for alanine, serum glutamate pyruvate transaminase. So SGOT, SGPT. You must have seen these words in the liver function test report, SGOT, SGPT, right? So when we do the transamination of the different amino acid, among them, the two are very important, aspartate and alanine. How they are converting into glutamate, what is the byproduct? We need to remember the byproducts. See, because in every transamination reaction, there are few things which are fixed. One thing which is very fixed is the product. Whenever you do the transamination, the product is glutamate. The byproduct is changing. One more thing which is fixed is the bisubstrate, alpha ketoglutarate. Whenever we do the transamination, we require alpha ketoglutarate. So we don't have to remember alpha ketoglutarate. We don't have to remember glutamate, because by default, they are going to come. So what we need to remember is the amino acid and the byproduct. So aspartate, the byproduct is oxaloacetate. How to remember? We use a mnemonic, a weird mnemonic, SOLP, and we will use it every day in every class of the biochemistry. And that is how we will remember this. SOLP. See, what is that? S stands for aspartate. Aspartate, when you do the transamination, the byproduct is oxaloacetate. In the same way, alanine, if you do the transamination, the byproduct is pyruvate. So it is going to be LP. Alanine, when you do the transamination, it's pyruvate. So LP. Aspartate converts into oxaloacetate. Alanine converts into pyruvate. So LP. So LP. Now, we understand that, okay, why these two reactions are important, the SGOT and SGPT, because they are the component of the liver function test. Now, here you can see the TPL. The threonine, proline, and lysine, they don't undergo transamination because they directly go to the liver and they will directly donate their ammonia to the liver. They don't undergo transamination. Now, how to interpret the liver function test? Although you will discuss it in more detail in the hepatology, but as of now, we'll get some basic idea. The liver diseases, we can divide into two categories: alcoholic, non-alcoholic. Whenever there is a liver disease, the SGOT, SGPT, they are going to increase. If it is an alcoholic liver disease, the SGOT will increase more. And if it's a non-alcoholic liver disease such as hepatitis B, hepatitis C, Wilson disease, or any non-alcoholic liver disease, the SGPT will increase more. So in alcoholic, OT increases more, and in the non-alcoholic, the SGPT increases more. But when the cirrhosis develops, that is the last stage of any liver disease, whether it's alcoholic cirrhosis or a non-alcoholic cirrhosis, whenever the cirrhosis will develop, the SGOT will be more compared to PT. Whether it's alcoholic cirrhosis or non-alcoholic cirrhosis, the OT will be more. Now, what is the significance of knowing this thing? That SGOT will be more. Now, you give me the answer. See here. No need to write this. What happened was, one of your relative is positive for, let's say, hepatitis B. In 2002, one of your relative has got the hepatitis B virus. And the doctor has said that every 6 months, you should undergo liver function test. So he was checking his SGOT, SGPT every 6 months. And obviously, it's a non-alcoholic liver disease. So SGPT was more compared to OT. And every 6 months, he was doing that. So now it was year 2025. And you reached to his home. And now what happened was, he has given his file to you just to check the file. When you looked at the report of liver function test, the SGPT was more till the year 2020. The SGPT was more. But after that, there was change in the trend. Now, the SGOT was increasing more than the PT. The OT was increasing more than the PT. The moment you see this, in a non-alcoholic liver disease, the SGOT has started increasing. In a non-alcoholic patient, in a non-alcoholic liver disease patient, if the SGOT has started increasing more than the PT, means this non-alcoholic liver disease has converted into what? Cirrhosis. It has converted into cirrhosis. So that is one of the hint that will be there in the question, that what is the significance of knowing the pattern of SGOT, SGPT. Now, what are the other components that we are having? SGOT, SGPT, it is whenever there is hepatocellular injury, the OTPT will increase more. Alkaline phosphate is a marker of cholestasis. Cholestasis means obstruction in bile flow. Whenever there is obstruction in the bile flow, the alkaline phosphate, the GGT, 5-base nucleotides, they are going to increase. They are the marker of cholestasis. The alkaline phosphate is not a specific marker actually. GGT and 5-base nucleotides, still they are specific for cholestasis, but alkaline phosphate is not, because alkaline phosphate can be increased in many other diseases. Actually, there are 5 tissues where you have alkaline phosphate. The mnemonic for that is BPL kit. Where the alkaline phosphate is found, it is found in the BPL kit. BPL kit. What is BPL kit stands for? BPL kit is bone, placenta, liver, kidney, and intestine. If there is a problem in any of them, the alkaline phosphate can increase. So alkaline phosphate, not specific. It's not specific. The mnemonic is BPL kit. Bone, placenta, liver, kidney, and intestine. But okay, now I have a question for you. You have understood what is SGOT, SGPT. But tell me that SGOT, SGPT, they are working where, in the liver or in the extrahepatic tissue? SGOT, SGPT, they are the enzymes of transamination, and the transamination is happening where? It is happening in the liver, or it is happening in the extrahepatic tissue? The transamination is happening in the extrahepatic tissue, right? Then why I'm saying that if there is a liver disease, SGOT, SGPT will increase. If they are the enzymes of the extrahepatic tissue, how they are related to the liver? How they are related to liver, and why only the SGOT increases in alcohol? Why not the SGPT? Likely asked the Srishti, right? So why such things are there? Giving you the explanation for that. See, here is the representation of the blood, and here is the extrahepatic tissue. For that example, let's say muscle. Actually, the enzymes are synthesized in the liver. The enzymes are synthesized in the liver, in the hepatocytes. In the hepatocytes, for example, let's consider this liver as a single hepatocyte, and here is the mitochondria of the liver. Now, there are the enzymes which are here in the cytoplasm. The SGPT is made, and the SGOT is made in the mitochondria of the liver. Mitochondria of the liver. Once these enzymes are made, these enzymes are going to transport to the blood, and then they will go to the extrahepatic tissue and will work there. So actually the enzymes are made in the liver, and then they are working in the extrahepatic tissue. But now what happens is, those who are consuming alcohol, we know that alcohol is a hepatotoxic substance. It is going to damage the hepatocytes, and alcohol is more prominently damages the mitochondria of the hepatocytes. So now when someone will consume the alcohol, because the alcohol metabolism is primarily happening in the mitochondria, so it is going to affect the mitochondria more. It is going to affect the cytoplasm also, but more the mitochondria, and they will rupture more, and these enzymes will come out more and more. So that is why they are going to increase in the blood, the SGOT, SGPT, whenever there is damage in the liver. If it is alcohol, it is going to damage more the mitochondrial enzymes. So the SGOT increases more, whereas the non-alcoholic liver disease, that damages the cytoplasm more. So the SGPT is going to increase more, right? I hope this doubt is clear now, that why the alcohol, the SGOT is going to be primarily increasing more, because it's a mitochondrial enzyme, and alcohol metabolism is going to happen in the mitochondria. Now, the next thing is the other correlations which are there. SGOT, it is just not found in the liver cells. SGOT is also found in the skeletal muscles. Also found in skeletal muscle. It is also found in cardiac muscle. So SGOT is also found in skeletal muscle. It is also found in cardiac muscle. It's found in liver cells. So SGOT is a non-specific one, right? That is why if there is rhabdomyolysis, in case of rhabdomyolysis, because in the muscles we have the SGOT enzyme, and if the muscles will rupture, a lot of SGOT will come out. So there is only going to be increase in SGOT, and the ratio of SGOT SGPT will be more than 5 in rhabdomyolysis. In myocardial infarction also, because in cardiac muscles we have the SGOT, so in myocardial infarction the SGOT is going to increase. In cirrhosis, whether it is alcoholic or non-alcoholic cirrhosis, the SGOT is going to increase. So there are some more conditions where the SGOT increases. It's just not the liver disease. It can be rhabdomyolysis, myocardial infarction, because in the muscles and the in the muscles, cardiac muscle, skeletal muscle, we have the SGOT. Normally, the SGOT, SGPT value is 30, 40 international unit. There are only few diseases where the SGOT, SGPT can go in thousands. Can you tell me the few diseases where the SGOT, SGPT will be in thousands, 5,000, 10,000, 8,000? Right? What are the few diseases where the SGOT, SGPT is in 1,000? One is autoimmune hepatitis. We will discuss in gastrology. The autoimmune hepatitis, the OTPT will be in thousands. Then, yes, it is drug toxicity. Drug toxicity can lead to OTPT thousands. It can be Wilson disease, ischemic hepatitis. And sometimes when the viral hepatitis becomes very aggressive, less likely said Kevin, it is fulminant hepatitis. So very aggressive viral hepatitis is a fulminant hepatitis. So these are the 5 conditions which you should remember when the SGOT, SGPT will be in thousands, more than 1,000 international unit, autoimmune, drug, Wilson, ischemic hepatitis, and fulminant hepatitis. So this is the first thing in the biosynthesis of urea. We convert all the different amino acid into glutamate. That is transamination. For us in the biochemistry, there will be 3 thumb rules. And the entire biochemistry, we will evolve around those 3 thumb rules. If we'll be good with these 3 thumb rules, there will be no problem with the enzymes. Most of the enzymes you can easily remember with these 3 thumb rules. The first one, in any reaction of biochem, if you see NAD, NADH or FAD, FADH, if you see these molecules, the name of the enzyme is going to be dehydrogenase. Dehydrogenase. In any reaction of biochem, if you see NAD, NADH, FAD, FADH, the name of the enzyme is going to be dehydrogenase. Let's take the example. The second step of biosynthesis of urea is oxidative deamination. What is oxidative deamination? The glutamate will reach to the liver. In the liver, from the glutamate will take out the ammonia. That is called as deamination. And in this reaction, NAD converts into NADH. And whenever there is NAD, NADH, what is the name of enzyme? Dehydrogenase. So it is going to be the glutamate dehydrogenase. Where this reaction is going to take place, the oxidative deamination is going to take place in the hepatic tissue. And in the hepatic tissue, where? Because it's an oxidative reaction. If you remember, oxygen is present in which organelle? In the cytoplasm or the mitochondria? Oxygen is present in mitochondria. So anything which is having oxidative word, they are going to happen where? In the mitochondria. Oxidative deamination will occur in mitochondria. Right? So this is the step number 2. That is very easy because the name itself was knowing, saying us, and we were also knowing that the second step is oxidative deamination. The glutamate which has reached to the liver will take out the ammonia. That is oxidative deamination. Now coming to the step number 3, that is the transport of ammonia. From the muscles, how we'll bring the ammonia? From the brain, how we'll bring the ammonia? Because the liver is the only organ which can convert the ammonia into urea. For example, we are having some ammonia molecules in the muscle and I want to bring it to the liver. In the muscles, for the energy, we consume glucose and we make the pyruvate. We will discuss that tomorrow. In the glycolysis, the glucose converts into pyruvate. But what is going to happen with this pyruvate? See, this pyruvate can be used to bring this ammonia molecule to the liver. The pyruvate and ammonia, they will merge together and they will make a molecule called as alanine. This alanine will come in the blood, and this alanine molecule will reach to the liver and we will take out the ammonia back from the alanine, and the alanine will bifurcate into ammonia and pyruvate back. So basically that who is the transporter of the ammonia molecule from the blood? It is going to be alanine. These reactions are not important. Just the word is important. That who is the transporter of ammonia from the muscle? It is alanine. We know that what will happen with these ammonia molecules? These ammonia molecules will convert into urea. That is fine. But what will happen with pyruvate? This pyruvate can convert back into glucose and that is called as the gluconeogenesis. And these glucose molecule will come back in the blood, will reach to the muscles, and the muscles will use these glucose molecules to do the glycolysis and generate the ATP. So this entire cycle is also called as the glucose-alanine cycle, or also called as Cahill cycle. Cahill cycle. But this Cahill cycle is not at all important. What is the single most important question is, who is the transporter of ammonia from the muscle? The answer is alanine. In the same way, if they ask that, who is the transporter of ammonia from the brain and the other tissues? How we transport that? And I have a question for you. In this one, you will tell me that answer. For example, here is the ammonia molecule in the brain, and I want to bring it to the liver. So what the brain will do is, this ammonia molecule will merge with the glutamate and will make a molecule glutamine. And this glutamine molecule will come in the blood. Via the blood, this glutamine will reach all the way to the liver. And from the glutamine, you will take out the ammonia. And this ammonia will go into urea cycle. It will go into urea cycle and will convert this ammonia into urea. But now you will tell me two answers. What is going to happen with this glutamate? Ammonia you will convert into urea, very good. But what you will do with this glutamate? What you will do with this glutamate? Can you take out the ammonia from the glutamate also? If you remember, what was the step number 2? From the glutamate, we can take out the ammonia, and that process is called as what? If you take out the ammonia from the glutamate, that was the step number 2. From the glutamate, if we take out the ammonia, yes, that is called as what? Yes, that is called as oxidative deamination. We have already did that, oxidative deamination. From the glutamate, we have taken out the ammonia. Yes, rightly said. And that ammonia will again go to the urea cycle. But the catch is, this some of the glutamine molecules goes to the kidney also. And they are also going to donate the ammonia in the kidney. So actually the glutamine molecules are going to the two places to donate the ammonia. One, mostly going to the liver and donate the ammonia in the liver, but some glutamine donates the ammonia in the kidney. But what this ammonia is doing in the kidney? One of the important function of your kidney is to throw out the acid from the blood. In the blood, for example, here is the representation of the blood. And in the blood, whatever acid molecules we are having, we want to throw them out. And which cell is doing that? How you eliminate the acid from your blood? In the nephron, we have a cell called as alpha intercalated cells. These alpha intercalated cells are going to merge this acid molecule with ammonia and will make the ammonium ion, NH4 plus. And this NH4 will be eliminated in the urea. So this ammonia is also helping us to eliminate the acid from the blood with the help of alpha intercalated cells. Now, the question is, if by chance these cells are not working, if they are defective, if the alpha intercalated cells are not working, the acid will not be eliminated from the blood and the acid will increase, the acidosis will happen. Can you tell me that this is called as which renal tubular acidosis? This is called as what? The renal tubular acidosis type 1, 2, 3, 4. If the alpha intercalated cells are not working, yes, very good, Pradeep, Aditya. It is called as RTA type 1. RTA type 1. If the alpha intercalated cells are not working, the acid will not go out, the acid will accumulate in the blood, and that is called as renal tubular acidosis type 1. So now we have understood that the glutamine is a very important molecule. It is transporting the ammonia to the liver from the brain and most of the tissue. But along with that, some glutamine molecule goes to the kidney and helps in eliminating the acid from the blood also. Right? So this is the step number 3. So we have completed the first 3 steps. Let's revise them. First step in the biosynthesis of urea. There are total 4 steps. The first step, you convert all the amino acid into what? Glutamate. Can you tell me that when you convert all the amino acid into glutamate, the name of the enzyme was transaminase. What was the coenzyme? B3, B5, B6. What was the coenzyme for transamination? B6. And what was one weird mnemonic that we have used? What was that one weird mnemonic? How the aspartate converts into glutamate, how the alanine, the weird mnemonic, SOLP, right? Then we did the step number 2. The glutamate has came to the liver, and from the glutamate, you are going to take out the ammonia. From the glutamate, when you take out the ammonia, that is called as oxidative deamination. Oxidative deamination. Then the step number 3, the transport of ammonia. From the muscle, the transporter is alanine. From most of the other tissue, it is glutamine. Now coming to the step number 4, because now the lever is loaded with the ammonia, we will run the urea cycle and will convert this ammonia into urea. That is going to be the last step. But before we go to the last step, let's do the second thumb rule for us in the biochemistry. The first thumb rule was whenever there is NAD, NADH, the name of enzyme is dehydrogenase. Now, the second one. For example, there are two small molecules are there. And if we join them and we make a bigger molecule, two small molecule joins and makes a bigger molecule, synthesize a bigger molecule, we have to see that whether we are using the ATP or the ATPs are not used. If you are synthesizing without ATP, the name of the enzyme is synthase. But if we are synthesizing with ATP, the name of enzyme is synthetase. So whenever we do synthesis, we try to see whether it is synthase or synthetase. So the step number 4, and here is a question for you before we really go to the step number 4. Step number 4 is very simple, that we convert the ammonia into urea. That is the step number 4. But the question is, what are the sources? What are the sources of ammonia you are having for your liver? One is the oxidative deamination of which amino acid? Can you tell me that? Which amino acid undergoes oxidative deamination and gives you ammonia? What is the name of that? Yes. Which amino acid? Glutamate. The glutamate. Can you tell me 3 amino acid which comes directly to the liver and gives the ammonia? Yes, that is what? TPL. They directly come to the liver and they donate the ammonia. Then we have the transporters of ammonia. Alanine is a transporter of ammonia from which tissue? From the brain or the muscles? Alanine is bringing the ammonia from where? It is bringing from the muscle. And the glutamine is bringing from the other tissue, including the brain. So these are the 4 sources of ammonia we have. And now we will convert this ammonia into urea, and this urea molecule will come in the blood. That is called as the blood urea nitrogen. So if they ask that urea cycle, what is the place where the urea cycle is happening? The place is going to be the liver. In the liver, if they ask where the urea cycle, is it occurring in the mitochondria or cytoplasm? Then you will say that the urea cycle total 5 reaction. The first and the second reaction occurs in the mitochondria, whereas the third, fourth, and fifth reaction occurs in the cytoplasm. So some reactions in the mitochondria, some reactions in the cytoplasm. When it comes to the organelle, few reactions in the mitochondria and few reactions in the cytoplasm. Mitochondria and cytoplasm. Now, this urea cycle is also called as the ornithine cycle. It is also called as the Krebs-Henselt cycle. Krebs-Henselt cycle or the urea cycle or the ornithine cycle is the same thing. Now we will see that why it is called as so and so, why it is ornithine cycle or why it is urea cycle. The idea is very simple. In urea cycle, we'll try to convert the ammonia into urea. So here is the urea cycle, here is the hepatocyte. We are going to start with one molecule of ammonia and ultimately we are going to make urea. This is the target. There are going to be total 5 reactions. Out of them, the first 2 reactions are in the mitochondria. The first reaction, you take one molecule of ammonia, one molecule of carbon dioxide, and we join them and we make a bigger molecule. In this process, we use the energy. So now you tell me that whenever you are synthesizing a bigger molecule with the help of energy, the name of enzyme is going to be what? Synthase or synthetase? Synthetase. In this process, we are using 2 molecules of ADP converting into ATP. The name of the enzyme is carbamoyl phosphate synthetase 1. Because we have made the carbamoyl phosphate, so the name of enzyme is carbamoyl phosphate synthetase 1. The full form is written below. It is not synthase, it is synthetase. Synthetase. Because we have made the carbamoyl phosphate, so it's called as carbamoyl phosphate synthetase 1. It's a very lazy enzyme. In fact, it is the most lazy enzyme of the urea cycle. And whichever is the most lazy enzyme of any cycle, that is called as the rate limiting step. So this is the rate limiting step of urea cycle, CPS1. It is the most lazy enzyme. It is so lazy that to activate the CPS1, we require NAG. The NAG is the activator. NAG will come and will activate the CPS1 and acetyl glutamate. Then this carbamoyl phosphate, what we will do is, in the mitochondria, we have a molecule ornithine. So from the carbamoyl phosphate, we'll take the carbamoyl group and we will transfer that carbomoyl group on the ornithine. And since we are transferring the carbomoyl group, so the name of enzyme is transcarbamylase. We are transporting the carbomoyl group, transcarbamylase, and we are transporting on the ornithine. So the name of enzyme is ornithine transcarbamylase, OTC. The full form is below, ornithine transcarbamylase. We are transferring the carbomoyl group on the ornithine transcarbamylase, and we make a molecule called as citrulline. Then this citrulline comes out of the mitochondria. And here comes a foreign particle which joins the urea cycle in the third reaction. And the name of that is aspartate. The aspartate is going to join the citrulline. When these two small molecules unites with the help of ATP, we are going to make a bigger molecule called as arginosuccinate. So the name of enzyme is going to be arginosuccinate synthetase, because we are making a molecule with the help of energy. And in this reaction, the triphosphate converts into monophosphate, means two phosphates has been consumed. This arginosuccinate is a very big molecule. It will split into two small molecules, arginine and fumarate. And when we do the breakdown, the name of the enzyme is arginosuccinate lyase. That will help us in the breakdown. That is the lysis. Then this arginine will further undergo breakdown. But this time with the help of water, we'll do the breakdown of the arginine. We are going to make the urea and the ornithine. And this is what we wanted. But this time we are doing the breakdown with the help of water. So the name of enzyme is arginine hydrolase. Arginine hydrolase. And we are going to get the ornithine and urea. And this urea molecule will come in the blood. When the urea comes in the blood, it is called as the blood urea nitrogen. And these molecules will be eliminated out. And what is going to happen with this ornithine molecule? This ornithine molecule will go back in the mitochondria and it will be reused. And because we are reusing the ornithine molecule in the urea cycle, that is why this urea cycle is also called as ornithine cycle. To enter the ornithine inside, we require a special transporter that is called as the SLC25A15. This 25A15 is also called as the ornithine transporter 1. This is called as the ornithine transporter 1, which is made up of a protein 25A15. And the ornithine goes in. So this is how the entire urea cycle looks like. There are total 5 reactions, 2 reactions in the mitochondria, 3 reactions in the cytoplasm. One more catch point is that the ornithine which is produced here, that is consumed, that is very good. But in our body, we have some other sources of ornithine also, which are going to be consumed here. So in our body, wherever the ornithine we are having, we can consume the ornithine here in the urea cycle. We can bring the ornithine here and we can use the ornithine molecules here. So in urea cycle, which ornithine we are using? One ornithine which is produced there, which we use them, but whatever is the other sources of ornithine we have, what we eat in the food also, that ornithine is also going to be consumed here. But now, what are the questions that they are going to ask? See, in biochemistry, there is one very particular thing that is there special about biochemistry, that the problem with this subject, there are so many cycles are there. And we feel that as a student, that how we are going to remember all these cycles in the sequence, that is the most difficult part. But the good thing is, no one is going to ask you that write down the urea cycle. NEET PG or NECT exam, no one is going to ask you, right? They will ask a certain set of questions from these cycles. After every cycle, I will tell you that what is the important points that you must remember. So in the biochemistry, you don't have to cram the cycles, that after the class, you are writing the urea cycle 50 times and trying to cram it. It's not going to solve the purpose, because eventually we are going to forget it. Instead of that, we should focus on what are the important key points that is there in the cycle, and we'll just focus on that part. For example, the first question they ask is, how much energy is used in urea cycle? In the first reaction, there are 2 ATPs which converts into 2 ADP. So I can say that the triphosphates converts into diphosphate, and we are using such 2 triphosphates convert into 2 diphosphates. So total 2 phosphates are consumed in the first reaction. In the third reaction, we are having 1 ATP, we convert into 1 AMP. So again, 2 phosphates are consumed here. So 2 phosphate in the first reaction, 2 phosphate in the third reaction. Total, how much phosphate is used? Total 4 phosphate molecules are used. But if they ask that total, how much ATPs are used? In the first reaction, how much ATP are using? 2 ATPs. In the third reaction, how much ATP are using? 1 ATP. So it looks like as if we are using 3 ATPs. But the best answer is not going to be 3 ATPs. The best answer is going to be 4 ATPs only. Why? Because the AMP which you are making, actually, the amount of the phosphate that you consume is equal to the number of ATP that you have used. Although it looks like as if we are just using 3 ATPs, but when you are having this AMP molecule and when you try to convert back into ATP, you will actually require 2 ATP molecules. So if they ask that how much ATPs are consumed here, the best answer is going to be 4 ATP. But if it is not given in the choice, then the second best answer is 3 ATPs. Because the number of the consumption of ATP is equal to the number of consumption of phosphate. Jitna phosphate use hua, the same amount of ATP has to be is consumed. We can say like this. What is the rate limiting step? The rate limiting step is the most lazy enzyme, that is the CPS1. Who is the activator of CPS1? NAG. Okay, now you tell me one answer. Total, how many reactions are there in urea cycle? Total, how many reactions are there in urea cycle? 5. Means there are 5 enzymes. Sometimes they ask that, what is the 6th enzyme of urea cycle? This NAG is considered as 6th enzyme of urea cycle. NAG is considered as the 6th enzyme of urea cycle. Then they ask that all of the following are cytoplasmic enzyme of urea cycle except, because you can see that the combination is like this. 2 enzymes are in the mitochondria and 3 in the cytoplasm. And they ask that which is the cytoplasmic enzyme, which is the mitochondrial enzyme, and how we will remember this. Yes, rightly said, Keval, that the cytoplasmic enzyme starts with the alphabet A. So if they ask that which are the cytoplasmic enzyme or all of the following are cytoplasmic except, then the answer is the enzymes which are starting with A, they are cytoplasmic. The remaining one is going to be the mitochondrial. So we'll remember like this. Which enzymes are cytoplasmic? Starts with A. The enzymes which starts with the A, they are the cytoplasmic enzyme. What is the formula of urea? The urea which you have made, it looks like this, NH2CONH2. But if you remember that, and you were listening very carefully in the answer that, when I said the first reaction of urea cycle, the first reaction of urea cycle, you started how much molecules of ammonia and how much molecules of carbon dioxide. In the first reaction of urea cycle, how much molecules of ammonia, how much molecules of carbon dioxide? One molecule, right? And look how the urea is looking, NH2, CONH2. So I can say that CO group is coming from the carbon dioxide. NH2 group is coming from the ammonia, which we have started with. But from where this second nitrogen is coming? Who has given this second ammonia molecule? In the urea formula, from where this second nitrogen is coming? There is one foreign particle which joins the urea cycle in the third reaction. Yes, rightly said everyone. It is aspartate. So they ask that who is the donator, who is donating the nitrogens in the urea? There are 2 nitrogens in the urea. One nitrogen is coming from ammonia and another nitrogen is coming from the aspartate molecule. Now coming to the last point in the urea cycle chapter, and that is the clinical part of the urea cycle. They ask that, okay, before I go to that, you tell me one simple answer. You tell me one simple answer. Urea cycle is converting the ammonia into urea. By any chance, if the urea cycle doesn't happen because of deficiency of any enzyme, the urea cycle is not happening. What will happen with the urea level? They will be more or less. If you don't do the urea cycle, what will happen with the urea levels? They will be less. And what will happen with the ammonia levels? They are going to be more, right? So whenever the urea cycle doesn't happen, the ammonia will not convert into urea and the ammonia increases, right? And that is called as hyperammonia. If you look at the urea cycle, see here, this is a simplified version of urea cycle. This is what we have discussed. There are total 5 reactions. We started with the first reaction, the ammonia and carbon dioxide. The first enzyme was CPS1. Then the second enzyme we have used the OTC. We made the citrulline. The citrulline came out, converts into arginosuccinate, then arginine, and then urea. And this urea will go into blood, making the blood urea nitrogen. By chance, if this first enzyme is not there, there is an individual, there is a child, he is having deficiency of this enzyme, CPS1. then this reaction will not happen. And if this reaction will not happen, the ammonia will not be able to convert into urea and the ammonia will increase. And this is called as hyperammonia type 1. Because the first reaction is not happening, so ultimately what will happen with the blood urea nitrogen levels? They are going to be less. So in any MCQ of biochem or pediatrics, if they say ammonia is more and the urea is less, means the urea cycle is not happening. It's a urea cycle disorder. If the ammonia is more and urea is less, it's a urea cycle disorder. If the first enzyme is not there, it is called as hyperammonia type 1. If the second enzyme is not there, what is the name of the second enzyme? The name of the second enzyme was OTC, ornithine transcarbamylase. If there is deficiency of this, then this reaction will not happen. The carbamoyl phosphate will increase, the ammonia will increase, and this is called as hyperammonia type 2. So in hyperammonia type 2, the ammonia will be more, the urea will be less. Then you must say that how you will differentiate the hyperammonia type 1 from type 2. Can you tell me, Keval, Syed, Ritika, can you tell me that how you will differentiate hyperammonia type 1 from type 2? In hyperammonia type 2, what is the other thing that is also more, which was not increased in type 1? What is that? It is the carbamoyl phosphate. In type 2, the carbamoyl phosphate is also more, right? And this carbamoyl phosphate, what happens is, because it is not able to convert into citrulline, because the enzyme was not there, the OTC enzyme is not there, so this carbamoyl phosphate will undergo some other reactions, and this carbamoyl phosphate will help in the synthesis of the pyrimidines. The pyrimidines such as the orotic acid, the CUT. If you remember in the DNA, RNA, you may have heard about the purines and pyrimidines. They are actually made up of, the pyrimidines are made from carbamoyl phosphate. So in this patient, the carbamoyl phosphate is more. So this carbamoyl phosphate will convert into the different types of pyrimidines such as orotic acid, which can convert into uracil. So these things will be more. So if they say hyperammonia, if there is more orotic acid, more uracil is there, then it is going towards hyperammonia type 2. So the hint for us in the MCQ is, if they say hyperammonia with orotic acid urea, if the orotic acid is also more and the ammonia is more, it is hyperammonia type 2. It's hyperammonia type 2. Let's go to the third one. Now, this is slightly tricky one. In the urea cycle, it is also called as ornithine cycle, because the ornithine which you produce, this ornithine is reused. This ornithine goes in the mitochondria with the help of ornithine transporter 1. This ornithine transporter 1 is also called as the SLC 25A15. 25A15. And the ornithine goes in. And I already told you that the ornithine which is produced in the urea cycle, that is consumed, but along with that, there are some other sources of ornithine is also there. The ornithine which you are eating in the food, all these ornithine is also getting consumed here in the urea cycle. But now what happens is, just imagine that if this transporter is not working, if this transporter is not working, the ornithine can't go in. If there is a defect in the ornithine transporter 1, if it is defective, the ornithine will not go in and the ornithine will increase. The ornithine will start increasing in the cytoplasm. Along with that, because the ornithine is not able to go inside, so the urea cycle can't work. And if the urea cycle can't work, this, because ornithine is not going in, so this reaction cannot happen. The ammonia is going to increase. The carbamoyl phosphate is going to increase, but they can't convert into citrulline because the ornithine is not going in. And this carbamoyl phosphate will convert into a special molecule that is homocitrulline. And the homocitrulline will increase. So now I have a question for you, that if you look at the second one, in that also the carbamoyl phosphate was more. And in that I said that carbamoyl phosphate will convert into orotic acid in the hyperammonia type 2. And here I am saying that the carbamoyl phosphate will convert into homocitrulline. Why I am not saying orotic acid? Can anyone tell that why I am not saying orotic acid here? Why the carbamoyl phosphate is converting into homocitrulline, not orotic acid? Because here in the carbamoyl, in this particular diagram, the ornithine is not there. But if you remember the name of enzyme, ornithine transcarbamylase, the transcarbamylase enzyme is there. the transcarbamylase enzyme converts the carbamyl into citrulline. But because the ornithine is not going in, but the enzyme is still there, the transcarbamylase enzyme is there, so it is going to transfer the carbamyl group on some other substrates. And instead of citrulline, we are going to make something similar to that, that is homocitrulline. Because we were having the enzyme transcarbamylase. So that will transfer the carbamyl group, and instead of citrulline, we'll make the homocitrulline. But here in the second example, the enzyme was not there. So the transfer of carbamyl group was not happening. So we were not able to make citrulline or homocitrulline. Nothing was made. And this carbamyl phosphate was converted into orotic acid in hyperammonia type 2. But here it is going to convert into what? It is going to convert into homocitrulline. So if the ornithine transporter is not working, actually there are three things are increasing. What are the three things increasing? One is homocitrulline, second is ammonia, and third is ornithine. These three things are increasing. And because there is three things are increasing, there is hyperammonia, there is hyperornithinemia, there is homocitrullinemia. This is also called as triple H syndrome. And the problem is this ornithine, if it is increased significantly, it is bad for the eyes. This ornithine, because it is increased, it is going to damage the eyes. It is going to damage the retina. And on the retina, you can see these white color patches will be there. That is called as gyrate atrophy. This ornithine is bad for the eyes. That will lead to damage in the retina, the retinal damage. And this entire disease is called as the triple S syndrome. So if the ornithine transporter is not working, it's called as triple S syndrome. Triple S syndrome stands for its acronym, that is hyperammonia, hyperornithinemia, and homocitrullinemia. Hyperammonia, hyperornithinemia, and homocitrullinemia. And don't worry about these urea cycle disorder. I will tell you the summary at the end, and you'll remember the entire the urea cycle there itself. Then comes the next one that is not very important. The third enzyme. Now you tell me the answer. The third enzyme was arginosuccinate synthetase. If there is deficiency of this, this reaction will not happen. If this reaction will not happen, the citrulline will increase, the carbamyl phosphate will increase, the ammonia will increase eventually. Because the citrulline is increasing, the third enzyme is not there, and because of that, the citrulline is increasing. So this is called as citrullinemia. It's not a very important disease. If the third enzyme is not there, it's called as citrullinemia. Now coming to the fourth one. If the fourth enzyme is not there, that is the arginosuccinate lyase. If there is deficiency of this, then this reaction will not happen, and this arginosuccinate will increase. There's going to increase in citrulline, carbamyl phosphate, that will also is going to happen. But because this molecule has increased, arginosuccinate, this arginosuccinate, when it will increase, it can come in the urine. And that is called as arginosuccinic aciduria. This molecule will come in the urine. Plus this arginosuccinate is very bad for the hairs. It is going to damage the hair. If the arginosuccinic acid is more, it will damage the hair. So this patient will have more ammonia. Along with that, this patient will also have damage to the hairs. If you see this word, they are talking about arginosuccinic aciduria. So what are the buzzwords? What are the important clues that is to be remembered? And that will most likely will give you one question, 1 mark in the exam. Now look at these buzzwords. Whenever there is urea cycle disorder, this summary is very, very important. Whenever there is urea cycle disorder, the ammonia will be more and the urea will be less. The moment you see this combination, you should think that there is some problem in the urea cycle is going on. But which problem? If there is no associated finding, it is called as hyperammonia type 1. If there is increase in the orotic acid is mentioned, orotic acid, uracil, it's hyperammonia type 2. And this is the most common, hyperammonia type 2. If there is increase in the ammonia, there is increase in homocitrulline, there is increase in the ornithine. Because of that, there is gyrate atrophy. This is a defect in the ornithine transporter. This is called as triple S syndrome, hyperammonia, hyperornithinemia, and homocitrullinemia. If there is a deficiency of the third enzyme, arginosuccinate synthetase, no problem. It's going to be citrullinemia. But if they say hyperammonia with hair defect, it's arginosuccinic acid urea. So the hair defects is going to be the buzzword for us. These are the four questions, but the most important for out of this is the orotic acid urea. The moment you see hyperammonia with orotic acid urea, it's hyperammonia type 2, the most common urea cycle disorder. In fact, a question comes, the ammonia is more, urea is less, and you are not getting any clue, go for hyperammonia type 2, because that is the most common word and most commonly asked also. There's going to be more ammonia and there's going to be more orotic acid. The last point about the hyperammonia, and it's very clinically relevant for us. See, right now, what your liver is doing is converting a very bad substance, ammonia, into urea. And this urea is being thrown out. In fact, liver is the only organ which have the capacity to convert the ammonia into urea because the liver is having the enzymes for that. And from where these ammonia molecules are coming from? Two. One, the protein which you are eating, from the metabolism of that, we are making the ammonia. And second, the intestinal bacteria, the good bacteria of your intestine, they are also making the ammonia. So there are two sources of ammonia we have. And we convert the ammonia into urea, no problem. But what happened was, there was a batchmate of yours. His name was Dharmendra. Dharmendra was a chronic alcoholic since his college life. He was drinking like anything. And because of the chronic alcoholism, his liver is significantly damaged. He developed cirrhosis. If a person develops cirrhosis, I would say that his liver is not working nicely, so he can't convert the ammonia into urea. And the ammonia molecules are going to increase. If the ammonia will increase significantly, these ammonia molecules will come to the blood. They are very toxic molecules. These ammonia molecules will come to the blood, and via the blood, these ammonia molecules will reach to the brain. If the ammonia molecules reaches to the brain, they are going to increase the osmolarity of the brain. And because of that, there will be swelling in the brain will happen. The water will be pulled in the brain, and there is going to be cerebral edema. The patient will develop the cerebral edema because the ammonia has reached to the brain, and that ammonia molecules are going to pull the water molecules, will lead to cerebral edema. And the moment the cerebral edema will happen, the patient will become drowsy. So those who are having liver cirrhosis, the ammonia will be more, and that is going to damage the brain. The problem is starting from the liver part and ultimately affecting the brain. This particular disease is referred, as rightly said, this is called as hepatic encephalopathy. It's called as hepatic encephalopathy. So what is hepatic encephalopathy is a disease of the liver. Because of that, the ammonia is not able to convert into urea. The ammonia will increase, will go to the brain and will damage the brain, will lead to cerebral edema. So Dharmendra will have cerebral edema. And this is called as hepatic encephalopathy. How they are going to present to us? Initially, these patients will have, the earliest symptom is going to be the altered sleep pattern. They will have altered sleep pattern, means they will not be sleeping in the night time. Instead of that, they will be sleeping in the daytime. Then the other things, they will have the change in the handwriting. There is going to be dysgraphia, change in the handwriting. They will become more and more drowsy. They will become more and more drowsy. And one day, if the cerebral edema will be significantly more, the patient will go into coma also. To classify this, we use a classification. Can you tell me that what is the name of that classification to say the stage of hepatic encephalopathy? What is the stage? How we decide the stage? The more the drowsiness, yes, that is called as the West Haven classification. We use the West Haven classification for that. When these patients will come to us, if you see an alcoholic patient is coming to you, he's looking drowsy, he's having altered sleep pattern, altered behavior, you can check the flapping tremor. You can tell the patient to hold the hands like this. You and me, we can do this. But these patients in hepatic encephalopathy, their hand will flap like this again and again. This is called as the flapping tremor, also called as the hysterexis. Flapping tremor or hysterexis. Can you help me with these answers? If we do the investigation in them, what will be happening with ammonia levels in Dharmendra? Because he's not able to convert the ammonia into urea. His liver is not working, so the ammonia levels will be more. What is going to happen with the blood urea nitrogen levels? The blood urea nitrogens are going to be less because he can't make the urea. And because this patient is having cirrhosis, I already told you that whenever there is cirrhosis, what will be increased more, OT or PT? What will be increased more? More. Among the OT and PT, which will be increased more? Yes, it is going to be SGOT, whether it's alcoholic cirrhosis or non-alcoholic cirrhosis. How you will treat this patient? You understood the problem, you tell me the solution. The problem is he's having more ammonia, and that is creating the problem. So this patient should eat more protein or less protein, because this ammonia is coming from the protein, right? So this patient should eat more protein in the treatment or less protein, because the protein is giving the ammonia. The ammonia is also coming from the gut bacteria. We don't want these bad bacteria, so we want to flush these bad bacteria out. And to flush the bad bacteria out, we can use the lactulose. That is going to increase the stool frequency and will flush the not the bad bacteria. It's good bacteria only, but as of now, they are working bad for the patient. So we are going to clear the bacteria from the intestine. In fact, we can kill them also. These bacteria can be killed by rifaximin. So we can give the rifaximin that can kill these bacteria. We have a phenyl butyrate, that is a ammonia binder that we can give, and that will bound the ammonia, and that is going to decrease the ammonia levels. What are the other things that we can do? Can you tell me that this patient is having cerebral edema? So what you want to additionally give to this patient? Because this patient is having the cerebral edema, to decrease the cerebral edema, what we are going to give him? Yes, we are going to give him the injection of mannitol. 6. The urea cycle is not happening. I want that somehow in Dharmendra, the urea cycle should happen. And there are two molecules in the urea cycle which comes from the outside and which helps the urea cycle. Rightly said, Aditya. There are two things which comes from outside and helps the urea cycle. Can you tell me what are these two molecules and supports the urea cycle? What are these two molecules? One is the aspartate and second is the ornithine. They comes from outside and they help in the urea cycle. They help in the urea cycle. So if I want to stimulate the urea cycle, I will give these molecules to Dharmendra so that the urea cycle will stimulate it. But you tell me that in humans, which type of amino acid we have, L type or D type? Which type of amino acid you have, L or D? In humans, which type? Yes, rightly said. We have discussed it is L type. So you are going to give the L aspartate and L ornithine. So in the market, we have the powders available of them in small, small sachets. So we are going to give L-ornithine and L-aspartate. The sachets of them are available and written as LOLA. L-ornithine, L-aspartate. L-ornithine, L-aspartate. LOLA. Yes, so this is how we will try to treat this hepatic encephalopathy patient. Now, one last question. In hepatic encephalopathy, we have the flapping tremors. Can you tell me some more conditions where you have the flapping tremor? One is hepatic encephalopathy. Can you tell me some more condition where you have the flapping tremor? Yes, there are three conditions that I want you to remember for flapping tremor. One is hepatic encephalopathy. Yes, rightly said, Manoj. It is the carbon dioxide narcosis, that is hypercarbia. And one is the uremic encephalopathy. So here, let's come to this part, the summary part. And there is one question is there towards the end of the summary. The flapping tremor, it is seen in three conditions. What are the three conditions? One is the hepatic encephalopathy. Then is the uremic encephalopathy. And third is hypercarbia, means CO2 narcosis. Carbon dioxide is blue. So this is all about the second chapter, that is the biosynthesis of urea. Biosynthesis of urea. Let's summarize this chapter and then we'll move to the next chapter. And after that, we'll take the break for the dinner. Now I'm writing, okay, let me add one page here. And this basic revision is again for you guys only. So try to participate as much as possible so that we will remember the things in the classroom itself. We have started with the biosynthesis of urea. In biosynthesis of urea, basically in our body, we have the different sources of ammonia which are there. Ammonia is a very toxic molecule. We take it to the kidney, we take it to the liver and convert this ammonia into urea. And this urea will come in the blood. Then it is called as the blood urea nitrogen. It reaches to the kidney and will be thrown out. Now, here is one question that if by chance the kidney is not working, this we have not discussed, just an extra point. If the kidney is not working, then the urea will not be eliminated and the urea will increase. And this urea can go to the brain and can damage the brain. So that is why those who are having kidney problem, in them the urea will be more and that will go to the brain and will damage the brain. That is called as which encephalopathy? Uremic encephalopathy. We have discussed hepatic encephalopathy. That was the liver is not working. In liver is not working, then the ammonia will be more. But if the kidney is not working, then the urea will be more, and this urea can also go to the brain and can damage the brain. Anyways, that is right now not the point of discussion. The uremic encephalopathy. Let's come to the biosynthesis of urea. There are 4 steps will be there. First, you convert all the different amino acid into which amino acid? Can you tell me that? There is only one amino acid which said that I will go to the liver and I will donate the ammonia. What is the name of that? Yes, rightly said, it is glutamate. Right? That is the transamination. That was the step number 1. But looking at glutamate, what the glutamate will do is you convert the different amino acid into glutamate. The glutamate will go to the liver and will donate the ammonia. Now, if you take out the ammonia from the glutamate, this process is going to occur in the presence of oxygen. So this is called as oxidative deamination. But there are 3 more amino acids which said that we can also go to the liver and donate the ammonia. Can you tell me 3 more amino acids? Yes, that is TPL, not IPL, TPL. They also go to the liver and donate the ammonia. Then there are some more transporters of ammonia is there from the muscles, from the brain. There are transporters are there. Who is the transporter of ammonia from the muscles? Yes, it is alanine. And from the brain, it is going to be glutamine. And then this ammonia molecule will convert into urea and will be thrown out. That is the urea cycle. In the urea cycle, there are total how many reactions? Total 5 reactions. Out of 5 reactions, the first and second is going to occur in the mitochondria and the remaining is going to occur in the cytoplasm. There are 5 questions from the urea cycle and you tell me all the 5 answers should be correct. All the 5 answers should be correct. Can you tell me that the urea cycle, what is the rate limiting step of urea cycle? OTC or CPS1? CPS1. CPS1. Who is the activator of CPS1, which is also referred as the 6th enzyme of urea cycle? NAG. NAG is the activator. That is the 6th enzyme of urea cycle. Total, how much phosphate is used in urea cycle? If they ask phosphate, it is 4. If they ask ATP, it is going to be 4. If the 4 is not given in the choice, then you can go for 3 also. Then comes the urea cycle disorders. If whenever there is urea cycle disorder, the ammonia will be more and the urea will be less. In any MCQ, if you see this combination, ammonia is more, urea is less, it's going towards urea cycle disorder. But along with that, you have to find the additional features. And I am writing 3 additional features and you tell me the diagnosis. If there is more ammonia, less urea, there is increase in the orotic acid also. Then what is the diagnosis? Hyperammonia type 1, type 2, it is hyperammonia type 2. If there is increase in the ornithine or there is gyrate atrophy is mentioned, gyrate atrophy, then this is what? Triple S syndrome. Triple S syndrome. And if they say there is a problem in the hairs, the hair defects are there, then this is arginosuccinic aciduria. Arginosuccinic aciduria. Right? So this is all about the biosynthesis of urea. Biosynthesis of urea. I understand you are tired. Yes, the attention span is leaking now, but somehow it's biochem, and on top of that, this is the first day, so it takes the time. Yes, we'll do leg open also. Don't worry, cable. We'll do that. So it takes time to get into the flow, but slowly, once you come to the flow, you start understanding that how the questions will come, because See, in this competitive exam, every one question is just not going to decide the rank, right? It is also going to decide that what you will be in the next 30 years, 20 years, 40 years down the line, in which branch you will be standing, right? That one question will decide that whether we'll go in medicine, or we will go into derma, or will be a radiologist, or will be anesthesiologist, or maybe what, right? So every question matters, and we don't know that from where that question is going to come, right? So we don't know. The moment we take a slight break here and there, we start checking our mobile, and just for that 1 minute, maybe one question can come from that, and that can decide so many things in our life. Right now, it's not the consequence that is going to happen tomorrow, so we take it very lightly. कि ठीक है यार, देख लेंगे, कल कर लेंगे, we'll do it, the recordings will be there, we'll do it afterwards, or this or that. But the point is, the tomorrow never comes actually. The If it happens today, it happens today. It's never going to be tomorrow. And the moment we delay it to tomorrow, or we take it lightly, the consequence will come maybe after 6 months, maybe after 8 months. That is bound to happen, right? So let's take it in more serious manner. And these are the easy marks. Out of these 13, 14, 15 questions, you can easily get more than 80%, 90% correct, compared to the effort if you put in some other subject. And to get 90% correct marks there, it takes a lot of effort. If you compare with biochemistry, the effort will be less, and the things will be easy for you, right? So it's just the way you have to be attentive in the classroom, and that is all is required. And when I ask a question, try to answer it, be it very easy, be it very difficult, maybe right or maybe wrong, doesn't matter, because the more we answer, it's kind of a repetition, one, and second, it's to keep you in the classroom. So uh, let's be like that, and the things will be easy for us. Now we have the small chapters. You don't have to worry. You will not be bored now anymore. So now let's come to the next chapter, and I want the more inputs from you now at this point onwards, because now we have got some basic idea. We'll have the dinner break around 7:45 till 8:15, and then from 8:15, 9:15, 10:15, and 10:45. Around 10:45, we'll wind up the class. The target is somewhere we'll try to complete the amino acid chapters today, and tomorrow we'll be doing the protein and the carbohydrate part. This is the outline. Maybe one chapter here or there can be, but more or less we'll try to finish like this. So we will have two sessions today, one before the dinner, one after the dinner, and in the same way tomorrow also will be the same. Now coming to the first individual amino acid, and now it is going to be more interesting. It's going to be more clinical now. Phenylalanine. What are the points that you already know about phenylalanine? Because the fan in the name, so it is aromatic. TV till 9 pmh, P is there, TV till 9 pmh, and that is essential, TV till 9 pmh. And there are four families which are water soluble. Can you tell me which are the four families which are water soluble? Acid, base, amide, and alcohol. These are the four families which are water soluble. Aromatic is not water soluble. Aromatic is not water soluble. This is water insoluble. Now, okay, one more answer then. One more answer. That there is one amino acid which starts with the T but non-essential. There is one amino acid which starts with the T but non-essential. Means you can make in your body. What is that? Tyrosine. Yes, rightly said. Tyrosine is non-essential because you can make in your body. How? See, phenylalanine is an essential amino acid. Means you must have to take in the diet. If you are eating phenylalanine, your body can convert the phenylalanine into tyrosine. So that is why tyrosine is non-essential. From phenylalanine, we can make tyrosine. How to do that? For that, we require the enzyme phenylalanine hydroxylase. And this enzyme only works if the BH4 is there. BH4. BH4 stands for the tetrahydrobiopterin. This BH4 will donate two hydrogens, and the phenylalanine will convert into tyrosine. But if I want to do this reaction again and again, I require again and again BH4. How I will get again and again BH4? So what I will do is again and again, the BH2 will convert into BH4. And for that, we will add two hydrogens. And in the 8th class, you have discussed about redox reaction. If you add oxygen to any molecule, oxidation. If you attach hydrogen to any molecule, it's called as reduction. So if I convert the BH2 into BH4, I have to add the hydrogen, and to add the hydrogen, the name of the enzyme is going to be BH2 reductase. Means technically what I can say is, if I want to do the reaction phenylalanine to tyrosine, actually I require two enzymes. To convert the phenylalanine to tyrosine, we require phenylalanine hydroxylase and we require BH2 reductase. If these two enzymes are there, we can do this reaction again and again, again and again. But by any chance, if any of these enzymes is not there, then this reaction will not happen. For that matter, let's see, we know that to convert the phenylalanine into tyrosine, we require phenylalanine hydroxylase and BH2 reductase. If there is deficiency of any of the one, either of the one is not there, then this reaction will not happen. And the phenylalanine can't convert into tyrosine. If the phenylalanine can't convert into tyrosine, the tyrosine will become less and the phenylalanine will be more. If the phenylalanine is more, it will stimulate the alternate pathway and it will convert into its ketone version. The phenylalanine will convert into its ketone version. And these ketone versions, such as the phenyl acetate, phenyl lactate, phenyl pyruvate, they will start coming in the urine. So if the reaction of phenylalanine is not converting into tyrosine, the phenylalanine will increase and will convert into the ketone versions. And these ketones will come in the urine. So this disease is called as phenylketonuria. If there is deficiency of any of the one, either hydroxylase or reductase, if there is a deficiency of hydroxylase, this is called as phenylketonuria type 1. Deficiency of reductase, it is called as phenylketonuria type 2, PKU. The full form is written below, phenylketonuria. So now you have understood that why, if these enzymes are not there, why it is not called as phenylalaninemia or why it is not called as hyperphenylalaninemia. Because the phenylalanine is increasing, but it converts into its ketone and they are more increasing and creating the problem. So this is called as phenylketonuria instead of phenylalaninnemia. And what are the ketones we have? We have the phenyl lactate, phenyl acetate, phenyl pyruvate. These are the ketones. These ketones are bad for the brain. So this child will have decrease in the IQ. There is going to damage to the neurons. Among them, this particular one, the phenyl acetate, it has a very bad smell. When this comes in the urine will smell like mousy, musty smell will be there. So it is because of phenyl acetate, the mousy smell of the urine. Plus, this child is having less tyrosine, and from tyrosine, we are supposed to make melanin. If the child is having less tyrosine, so less melanin synthesis, and if there is less melanin, the child will be fair. So actually, there are three symptoms in this child. What are the three symptoms? If there is phenylketonuria, the child will have fair complexion, decrease in the IQ, and bad smell in the urine. But you know what? The mother will never bring the child because of the first complaint. The mother is actually happy. The child is fair. The mother is actually happy. So the mother will never bring the child for fair complexion. The mother will bring the child because the mother will say that, sir, whenever I'm changing the diaper of this child, it has a very bad smell in the diaper. It's a mousy smell, musty smell that is there. And this is how these patients will come to you, because decrease in the IQ also will be presented in the later part of the life, right? After 2 years, 3 years of the age, then we'll be able to see that the IQ of the child is less. Initially, it will not be appreciated. So the fair skin complexion, the mother will never bring the child because of that. The decrease in the IQ will present after 1 or 2 years of age. Then the mother will understand that, okay, the child IQ is relatively less. It looks like this. So the one and the first and the only presentation symptom is going to be the musty smell of the urine. That is because of phenyl acetate, right? So here it is written how the phenylketonuria is going to present to us. These children will have the problem in the brain. So there's going to be mental retardation, seizure. There's going to be hyperactivity in the child will be there. These children will be failure to walk will be there. But one more interesting point is when this child is there in the fetal life inside the uterus. At that time also, the child was having the phenylalanine and which was supposed to convert into tyrosine. And after the birth also, he's supposed to convert the phenylalanine into tyrosine. But what was happening was this reaction, even the child is not having the enzyme, even in the fetal life, the child is able to do that because the phenylalanine to convert into tyrosine, we require the two enzymes, hydroxylase and reductase. If the fetus is not having the enzyme, the maternal enzymes will do the reaction for the fetus. And there is no problem in the fetal life. The mother enzyme will do the things. But once the child is born, after 72 hours, the maternal enzymes will be washed out. because the half-life of the metal enzyme is 72 hours. After 3 days, the child has to produce its own enzyme. But now the problem is the child is having enzyme deficiency, and then this reaction will not be able to happen. And then the phenylalanine will start increasing and the symptoms will come. So they ask this question that this child will have the symptom on the first day of the life. No, the symptoms will come after 72 hours. After 72 hours, this child is absolutely normal at birth. No problem at birth. The clinical feature will occur after 72 hours. And this child will have the mousy smell is due to what? Phenylalanine, or it is phenyl acetate? It is phenyl acetate. Phenyl acetate. There is profound mental retardation will be there, and this child will have fair skin complexion because of less melanin. You might not have came across any child in India or in your relative, not India, in your relatives that is diagnosed with phenylketonuria, because we don't routinely test for this disease actually. But in United States, every child has to undergo for the screening of phenylketonuria. What they do is, at the age of 72 hours or 96 hours, means after the third day of the life, they are going to prick the heel of the baby and they will take few drops of the blood. And in that few drops of the blood, this is called as the heel prick test. And they are going to check the enzymes, the phenylalanine hydroxylase, the reductase, whether they are there or not. This is called as the heel prick test. But in India, we don't do this routinely. But in US, it's mandatory for every child to undergo this. What are the other tests that we can do? We can do Guthrie test. The investigation of choice is going to be tandem mass spectrometry. We can do ferric chloride test also, but that is non-specific. What is tandem mass spectrometry? See, in a patient of phenylketonuria, we know that this reaction is not happening. So the phenylalanine is more and the tyrosine will be less. The tyrosine will be less. So we take the blood sample and we see that how much phenylalanine and how much tyrosine is there. And that is called as the tandem mass spectrometry. Like this, we are going to put the sample in this, and this machine will analyze how much phenylalanine, how much tyrosine in the sample. Normally, if you see, normally the phenylalanine is this much and tyrosine is this much, more or less same, or rather tyrosine is little more compared to phenylalanine. But in phenylketonuria, look at the phenylketonuria and look at the level of phenylalanine and compare with tyrosine. The tyrosine is very, very less compared to phenylalanine. And this is how we diagnose. In fact, this image itself can come and they can ask the diagnosis, right? If they give you this image stating that the phenylalanine is more and the tyrosine is less, it is going towards what? It is going towards the phenylketonuria. This is the report of tandem mass spectrometry, right? How to treat this child? How to treat this child? See, we need to understand the problem, and you will tell me the solution. The problem is this child is not able to convert the phenylalanine into tyrosine. And from where this phenylalanine is coming is from the diet. And because he's not able to convert this, so this phenylalanine is converting into some other things, the ketones, and creating the problem. So we have to give a food which is having less phenylalanine, almost no phenylalanine. So the problem will not happen. Plus, this child is having less tyrosine, so we have to give tyrosine supplement. So we have to give a formula feed, avoid phenylalanine in the diet. We need to avoid phenylalanine in the diet, and we need to supplement tyrosine. Need to supplement tyrosine in the diet. Right? This is the phenylketonuria. It is an autosomal recessive disorder. In fact, in biochem, if I don't tell you the inheritance pattern, it is by default, it is autosomal recessive. If nothing is written to you, it's automatically autosomal recessive, right? There are only few diseases which are X-linked and autosomal dominant, and I will tell you specifically about them. But if I'm not saying anything, by default, biochem is autosomal recessive disorders. So here it is asked that what is the inheritance pattern of phenylketonuria? It's autosomal recessive. The other answers you can write down on your own. It is due to deficiency of which enzyme, the mousy smell is due to what, skin complexion, and like this. Now, okay, now you tell me one answer. Normally, the tyrosine is essential amino acid or non-essential amino acid? Tyrosine is essential or non-essential? Tyrosine. Tyrosine is non-essential, right? Because you can make it from phenylalanine. But now you tell me that, does this child can make the tyrosine from the phenylalanine? Can this child make the tyrosine from phenylalanine? No, right? This child can't make the tyrosine, means in this child, the tyrosine has to be supplemented. So if they ask that in which disease the tyrosine becomes an essential amino acid you have to take in the diet, then it is going to be phenylketonuria, right? So let's write down this point that in this disease, in phenylketonuria, tyrosine becomes an essential amino acid. In phenylketonuria, the tyrosine becomes an essential amino acid. Okay. The next individual amino acid we are discussing is tyrosine. Just 5 minutes, 5 to 10 minutes more, and we'll take a break. When it comes to tyrosine, what are the points that we know about tyrosine already? Tyre, so it's aromatic. And since it's aromatic, so aromatics are water insoluble. When it comes to the essential, non-essential, we already know tyrosine is non-essential. This is the fifth time we are discussing tyrosine is non-essential because it can be made from phenylalanine. So it is a non-essential amino acid. And all the aromatics are mostly keto plus glucogenic. So these are the first 4 or 5 points, which you already know from the first chapter itself. So I don't have to go into detail of these 4 points. The special point about the tyrosine structure is it is having a special ring. In the tyrosine, we have the phenol ring. You don't have to remember the structure. Just remember that in tyrosine, we have a ring. And what is the name of that? Phenol ring. Phenol ring is seen in tyrosine. What are the special products we are making from tyrosine? There are 3 special products we are making. One is melanin, that is deciding the color of the skin. The second is the thyroid hormone, T3, T4, and the last is the catecholamines, such as dopamine, norepinephrine, epinephrine. This is what we are making from tyrosine. So these are the 3 special products that we make from tyrosine. And whenever there's a combination of 3, it's a potential all except quotient. Starting with the first one, the melanin synthesis. How the tyrosine converts into melanin. When the tyrosine converts into melanin, it's a very long reaction. We don't have to remember all the enzymes, just the rate limiting step. The rate limiting step is tyrosinase. And to do this reaction, we require copper also. This reaction is going to occur in the stratum basale of epidermis. Because in the stratum basale, we have the melanocytes. The melanocytes are going to make the melanin by converting the tyrosine into melanin. Tyrosine converts into melanin with the help of the enzyme tyrosinase and copper. But what if this enzyme is not there? If someone is not having tyrosinase, he can't make the melanin. And if there is no melanin, the skin will be completely white like this. You may have seen such many of these such patients, right? Complete head to toe, complete white. They are having the deficiency of tyrosinase. Please correct the spelling. They are having the deficiency of tyrosinase, not tyrosine. If there is deficiency of tyrosinase, the patient will be completely white, complete fair complexion. This is called as albinism. In albinism, the patient will be completely white from head to toe. But this albinism should not be confused with a similar disease where you have the hypopigmented patches, but not the complete from head to toe. If complete head to toe is white, it is albinism. It is due to defect of tyrosinase. That is fine. The tyrosinase enzyme is not there. But if there is white patches are there, then this is not albinism. This is vitiligo. And this vitiligo is basically because of the wrong antibodies against the melanocytes. If there is some wrong antibodies are made and they are destroying the melanocytes, that is called as the vitiligo. So in vitiligo, we have the patchy white area around the oral cavity, on the fingertips, on the axial part of the body. That is the vitiligo. But in albinism, there is no melanin at all. In vitiligo, the melanin is there, but the wrong antibodies are destroying the melanocytes and creating the problem. The last point before the break, it is the thyroid hormone synthesis, the T3 and T4. And this will help us just not in biochemistry, it will help you in endocrinology, it will also help you in pharmacology and physiology. How the thyroid hormone is made. See, in the thyroid gland, we are making mainly 2 hormones. One is the thyroid hormone and second is the calcitonin. And who is making that? If you look at this diagram, you can see some yellow colored cells and then you can see some tire-like structures. These yellow colored cells are the parafollicular C cells. because they are around the thyroid follicle. So they are called as parafollicular seed cells, and that is making the calcitonin. Calcitonin. Whereas these black structures which you can see, the tire-like structures which you can see, these are called as the thyroid follicles. And if I take out one thyroid follicle, you can see that in one thyroid follicle, this is the entire thing is the thyroid follicle. There are three things that you can see. The outermost, the red color thing is the blood capillary. Then the black color cells arranged in the circle that is called as the follicular cells. And in the center, there is some liquid is here in the center. And this liquid is called as colloid. So this combination of these three, the blood capillary, the follicular cells, and the colloid, this combination is called as the thyroid follicle. Now, this thyroid follicle is the one actually which is making the T3 and T4 and how it is working. So to understand this, let's take a small segment from here and we'll zoom in this area to understand that how the thyroid follicle is working. So the orientation of the diagram should be very clear. Now we are zooming in that diagram and there will be three things. What are the three things? Blood capillary, the outermost, then the follicular cells, and then the colloid. These are the three things. How it actually works. Now, the iodine which you are eating in the diet, these iodine molecules are absorbed in the intestine and they come in the blood. And these iodine molecules will try to enter in the follicular cells. From the intestine, it will come in the blood, it will go to the capillaries of the thyroid and it will try to enter in the follicular cells. To enter in the follicular cells, there is one channel that is called as the sodium iodine channel. And it is not open always. To open this sodium iodine channel, we require a special hormone that is coming from the anterior pituitary and that is TSH. The TSH will come and it is going to open the sodium iodine channels. And now the iodine influx will happen. Yes, it is a sodium iodine import that is happening, import that is happening. And these iodine molecules now will be pushed forward towards the colloid. To push in the colloid, we require a special protein and that is called as the pendrin protein. There is a special channel which is there, which is connecting the follicular cell with the colloid pendrin. And now the iodine has came in the colloid. Here in the colloid, we are going to make the couple of the iodide. And these couples of the iodide will made with a hormone called as, with the help of enzyme called as TPO. The full form is written below, thyroid peroxidase. We are going to make the couple of iodine that is called as iodide. And a lot of iodide molecules will make. When all this program is happening, meanwhile, the follicular cell has made a protein and the name of that protein is thyroglobin. What is so special about thyroglobin? That it contains a lot of tyrosine amino acid. Since it's a protein, so it's made up of amino acid, and which amino acid we are interested in is in tyrosine. It contains a lot of tyrosine. And now this thyroglobin protein will come in the colloid. And because it contains a lot of tyrosine, so the iodides which you have made, the couples of the iodine which you have made, the iodides, these iodide molecules will start attaching on tyrosine amino acid. On some tyrosine, one iodide will attach, and on some, two iodides will attach. If one will attach, it's called as monoiodotyrosine. If two will attach, it's called as diiodotyrosine, MIT and DIT, monoiodotyrosine and diiodotyrosine. If one will attach or two. Now this MIT and DIT, they will come back towards the follicular cells. And when they will come back, the mono can club with di, that is called as T3, or the di can club with di, that is called as T4. And this is how we make the T3 and T4. Mainly the things which is made is, it is going to be the T4. Mostly we are going to make the T4. It is inactive hormone and this T4 will be coming in the blood. It will be released in the blood. It is inactive. Now you must be thinking that why we are making an inactive hormone, why we are not directly making the active hormone, and why it is important. Because see, what happens is your entire body requires the thyroid hormone, be it bones, muscles, ligament, brain, heart, kidney, everyone wants thyroid hormone. So what the thyroid gland will do is it will make the T4, which is inactive, it will put in the blood. And it will tell the tissue that whoever wants, however you want, how much you want, you take the T4 from the blood and convert into the active hormone that is T3. And how to do that? This T4 comes in the blood. For example, the brain wants the thyroid hormone. The brain will take the T4 from the blood, will remove one iodine, from the fifth position. So the name of enzyme is 5-deiodinase. If you remove one iodine, with the help of the enzyme deiodinase, the T4 converts into T3, and the T3 is the active hormone. To do this reaction, we also require selenium. So selenium is helping us in converting the T4 into T3. And in fact, this is an important clinical question. If there is deficiency of selenium, which is seen in the mountain waters, particularly in the Himalayas, in the Himalayan water, the selenium is very less. So those who are drinking that water, they can't convert the T4 into T3, and in them the T3 will be less. Means the active hormone will be less. And if the active hormone, the T3 is less, means these patients will develop hypothyroidism. So selenium deficiency can lead to hypothyroidism because we will not be able to convert the T4 into T3, and that is particularly seen in the Himalayan water. So what we have understood about the thyroid hormone, the T3 and T4, one, it is mainly made up of tyrosine, MIT plus DIT or DIT plus DITs, right? So this MIT or DIT, basically they contains tyrosine. That is why we are discussing this topic here, MIT, DIT, the tyrosine ones, right? So, and what other things is, if you compare them, T4 and T3, the production wise, the T4 production is more, the T3 production is less. But if you see the activity wise, the T4 is less active and T3 is more active. The half-life of T4 is around 7 days, whereas the half-life of T3 is 1.5 days. And what is the relation with pharmacology? If a person is having hyperthyroid, how you can treat that patient is, you don't want the thyroid hormone to be made anymore if a person is having hyperthyroid. And how you can stop that? Now everyone see here, and no need to do the things in your diagram. Just see here, everyone. If a person is having more thyroid hormone and you want to decrease the thyroid hormone, there are three methods. One, you can stop the TPO. If you stop the TPO, the thyroid hormone synthesis will not happen. Or you can stop the release of T4. Again, the T4 will not come out, the hyperthyroid will be treated. Or we can stop the conversion of T4 into T3, that is the 5-deiodinase inhibitor. So there are three methods by which we can treat hyperthyroid. Either we can stop the TPO, or we can stop the release of T4, or we can stop the conversion of T4 into T3. There are three methods by which we treat hyperthyroid. And what are these drugs? The TPO inhibitors. The TPO inhibitors are thiamides, such as carbimazole, methimazole, propylthiouracil. Carbimazole, methimazole, the PTU. Then we have the thyroid hormone release inhibitor, that is the Lugol's iodine. If we give the Lugol's iodine, the T4 release will not happen. And then we have the 5-deiodinase inhibitor, such as the beta blockers, steroids. In fact, the thiamides also. So actually, thiamides are having two mechanisms of action. The thiamides stop the TPO as well as they stop the 5-deiodinase. The last point before the break, during the thyroid hormone synthesis, there was one protein which was pumping the iodine from the follicular cell to the colloid. If you remember that, what was the name of that protein who was pumping the iodine from the follicular cell? Yes, from the follicular cell to the colloid, the iodines were going via the pendrin channels. These pendrin channels, actually, these pendrin protein are found in two places in our body. One is the thyroid gland to make the thyroid hormone, and second is in the ear. So by chance, if there is a child and his pendrin is not working, the pendrin channels are found at two places, the thyroid gland and ear. If there is a defect in the pendrin channel, the thyroid gland will not work, means this child will have congenital hypothyroidism, and the child will also have the hearing loss because the pendrin channels are required in the ear also. So if a child is having congenital hypo with hearing loss, there is a means it is a defect of pendrin channel. It's called as Pendred syndrome. Called as Pendred syndrome. Can you tell me that Just a question from pediatrics. It's just an extra point. What is the most common cause of congenital hypothyroidism? What is the most common cause of congenital hypothyroidism? The options are Pendred syndrome, uh, iodine deficiency, selenium deficiency, Hashimoto thyroiditis. What is the most common cause of congenital hypothyroidism? Iodine deficiency, selenium deficiency, Hashimoto thyroiditis, Pendred syndrome. What is the most common cause? The answer is yes, one of them. One of you have given correct answer. It is none of them. It is the thyroid agenesis. The thyroid gland is not there by birth is the most common cause of congenital hypothyroidism. Congenital hypothyroidism, thyroid agenesis. Yes. So this is all about the thyroid hormone. So we have discussed from tyrosine, we are making two special things. One is the melanin that is deciding your skin color. And the second we have discussed from the thyroid, we are from the tyrosine, we are making the T3, T4. The third thing that we are going to make from the tyrosine is the catecholamine. That we'll discuss after the break, that how we'll make the catecholamines from the tyrosine, right? So it is 7:48. We'll take a break and we'll start at 8:20. Yes, Christine. We'll share this one with you. We'll be sharing this annotated one with you. Yes, the link will be open. So it's better you don't leave. Otherwise, you can rejoin also. It's up to you.